saliniferous soils, soils which are rich in selenium, by far exceeding that concentration. Now, if we take a look at the upper right-hand corner of this slide, there is a picture depicting the selenium cycle in the environment. So you can see that the predominant forms are the selenate, containing selenium in the 6th oxidation state, and selenite here. And there has been reports that selenate accounts for almost 67% of the total selenium concentration in the environment. So these forms of inorganic selenate will be uptaken by algae and other microorganisms, transferred higher in the trophic chain, and it is through these algae that the selenium cycle will continue through the methylation and conversion of selenium to a volatile species. So it is of high interest to us to examine, to investigate how these algae will respond to exceeding concentrations of rising concentrations of selenate of inorganic selenium in the environment. And for this purpose, we have taken our model algal organism, that is Chlamydomonas reinhardti, and cultivated in growing concentrations of selenate, selenium in the 6th oxidation state. So for this system, we have applied ICP-MS in order to determine the selenate uptake within the cell. We have also conducted size exclusion chromatography experiments to determine how much of that selenium is incorporated into proteins once selenate enters the cells, and also carried out some lipidomic studies in order to see what is the effect of selenate on the cell membrane lipids. And once we have exposed our Chlamydomonas cells in selenate, here we present growth curves, which depict the growth of each cell line in the different exposure media. So you can see here for the control and the other colors representing the selenate exposure concentrations. So blatantly at first, by looking at this graph, we can see a progressive lag growth, a very intensive lagging growth. So for example, if we would take the 5th day here, which is the 10th time point, we can see that the 20 micromolar incubation concentration has achieved almost 50% of the respective growth of the control, while the others reach roughly at 20%. So the first indication that cells are under significant stress. And if we were to examine our cells visually under the microscope, we could get some pretty well defined photographs. So for example, for the control, we can see a pretty good state of the cells with a distinct typical green color for these algae and a range of diameters in the acceptable range. Well, when we go to the 20 micromolar here, you can see that there is an increase in the diameters, so an indication of cell bloating, and also some aggregation here. Well, when we go to the extreme situation of the 150 micromolar of selenate, we get quite a big increase in the diameters here, extensive aggregation, and also kind of a distorted cell physiology. So these are proofs that cells are under significant stress. And in order to determine our uptake, we present here a brief description of our experimental setups. So briefly, we take defined portions from our cell cultures. We digest it with nitric acid, and once we have the digest and diluted, we introduce it to our ICP-MS. And we have carried out this experiment for unwashed cells, which are we are taking the amounts from the culture directly, but also for cells which have been, prior to digestion, treated with EDTA once or twice, washed with EDTA in order to see whether there is any selenate randomly bound on the cell wall. And these samples were introduced to our ICP-MS, which is a very sensitive instrument for allowing us to quantitatively determine metals in biological samples. So we get our signal here for selenium, and we can correlate the signal to selenium concentration in the cell digest that we have. And provided that we know here the cell concentration of our samples, because we have measured them visually under the microscope with counting calculations, this ratio can be expressed as selenium content per cell. So quantitatively here, we have the selenium content per cell expressed as femtograms per cell, as opposed as a function of the selenium incubation concentration. And you can see here the blue line depicting the unwashed series that is proportionally increasing as a function of the concentration. And this is also the case for the washed cells. Well, there is a big difference between the washed and the unwashed, especially for points above 50 micromolar. And this is the indication of the removal of the selenate, which has been randomly bound on the cell wall. So the gray line here would represent the selenium that is internalized in the cells because they are washed twice. So it's only the selenium that's inside the cell. and you can see that they are really governed with by a linear trend, as the high degree of linearity thus shows. And this is our experimental setup in order to investigate how much of that selenate has been biotransformed into protein-bound selenium in Chlamydomonas reinhardti. So briefly, again, we're taking defined portions of our culture and we suspend the cells in a lysis buffer. So here our target is to lyse the cells, break them, disrupt the membranes in order to extract the cytosol out. And we bring it to our sonication probe. So we do sonication cycles. And after that, we centrifuge the sample in order to separate the cell debris, the cell fragments from the cytosol, that is the supernatant. And as soon as we collect the supernatant, we introduce it to our size exclusion chromatography ICP-MS combined system. And if we analyze our samples, we get chromatograms looking like this. So we have distinct colors for color codes for the different treatments. And for example, if we take the control, the orange, we see no visible, no discernible peaks. Well, the treatments show clearly two visible peaks, two major peaks, and also a third one looking like a shoulder here to the second peak. So if we run inorganic selenium standards, we see that this peak identifies as selenium ionic. So this should be, the first one should be here, the protein bound, as is the case for protein macromolecules eluting from size exclusion chromatography columns. They are always eluting earlier. And so the existence of that third peak, we don't really know when it's work under investigation, but what we can speculate is it concerns selenium species of higher molecular weight than the selenium ionic here, for sure, but maybe close to the fractionation range of our column. So it could be metallothioneins or phytogelatins, small peptides that exist in the Chlamydomonas and have around the same molecular weight. And if we want to examine that quantitative, what we can do is from the intensity, if we integrate that peak here, which denotes the selenium protein bound, we can get the equivalent selenium concentration for the protein bound. So we can see here, this is depicted as a green one. And for the ionic, if we convert it to concentration, this is depicted as the blue line. So we can see that for the selenium protein bound, it's proportionally increasing as a function of the selenium incubation until 100 micromolar, where it levels off. And this is probably a saturation mass for the cells. And the addition of these two selenium species here should give the total selenium concentration in the sample, which we expect to be in agreement with the total selenium that we measure with the conventional ICP-MS, the first technique. And as you can see, we get an excellent agreement. Now, moving on to our lipidomic studies, a brief description of the setup here. We have our nebulizer, which with a flow of high pressure molecular nitrogen, we create a jet. We create a spray from our solvents. And what it happens is the droplets of that spray impart the surface of the cells, which, by the way, we have deposited them here as 1 microliter spots. And what really happens is the droplets, they extract the lipid molecules and convert them to the, and once they are converted, these lipid molecules in the gas phase, they get ionized. So these ions are collected then by the mass spectrometer and we get characteristic spectrum. Corresponding to some membrane lipid classes here that we can identify through fragmentation patterns and so on. So with that technique, the problem is we cannot achieve absolute quantitation, but what it allows us to do is we can look at discernible peaks through these lipid classes here, which denote the saturated versus the unsaturated form of these lipid acids connected to the triglycerol bone of the membrane lipid. So we can take plots of the ratios of that. So you can see here we have focused on the phosphatidyl glycerol membrane lipid, which is has 1 double bond in the saturated form and 4 double bonds in the unsaturated. So this would denote 3 double bonds multiplied by 2 hydrogen, a 6 Dalton difference in the mass spectrum. So the saturated versus the unsaturated form. And in order to visualize this data more clearly, we can plot them as box plots here. So this is the plot, the ratio of the saturated versus the unsaturated here. So an indication of the saturation degree. You can see here how the control levels are and how the selenate incubate grown cells here. The cells which have grown in selenate, they demonstrate an increase in this saturation degree. And I would like to show you an equivalent graph for Chlamydomonas incubated in selenite here. which there is no observable increase. It's just fluctuating over the control, around the control. Um, and again, this is another lipid class, sulfokinovosyl diacylglycerol, where we have the saturated and the unsaturated form. And again, the same plots, we see an observable and tremendous increase in the intensity ratio, 3 to 4 times for the cells which have grown in selenite, while for selenite, there is no observable increase. So in order to extract some conclusions out of this work, we have demonstrated a combinatory approach for allowing us to study the uptake of selenate, the fate of selenate once it enters the cells, whether it is protein bound or not, and also the effect that it has on the cell membrane lipids. Exposure to increasing concentrations of selenate demonstrated a progressive lag in cell growth. Selenate uptake increased linearly with increasing exposure concentration. Selenium is internalized by the cells. This was blatant, but is also randomly bound in the cell wall, and thus the difference between the unwashed and the washed cell line series. And also part of the internalized selenium is shown to be protein bound. And again, exposure to increasing selenate concentrations induced a reorganization to at least two classes of membrane lipids, which were characterized by a higher degree of saturation. And this is an indication of this translates to less fluid membranes. This is to acknowledge our companies and institutions for providing us funding to realize this work. And I would like also to thank you for your attention.
Thank you, Mavrakis, for your fantastic presentation and very nice work. I cannot see any questions from the audience, at least when I saw the link. I don't know if you can ask any questions. Is it any question?
Sorry.
Is there any questions from the
I stop
audience?
sharing.
Okay, thank you. Well, I have some questions because you showed that part of selenium is bounded to proteins. Is okay. Do you think that this selenium, which is bounded to proteins, can, because it is bounded to proteins, which I don't know, but probably they are not labile complex, they are inert complexes. Uh, can they be toxic or not inside the cells? Uh, do any assays in order to evaluate its toxicity or not yet? Do you intend to do it?
I think I have understood the question. And well, you could see from the growth curves that there was a blatant cell toxicity that selenate induces, um, showing by the lag in the growth, and also the
Yes.
photographs, which have altered physiology. Now there are reports in the literature reporting on the mechanism for toxicity of selenate in the cell. So there is could be a way that selenium, when the concentration is lethal to the cell, which is above the toxicity level of the EC50 and so on, could be incorporated into like, because there is too much selenium going into the cell, it is incorporated into the equivalent cysteine level, cysteine and amino acids. So exchanging for sulfur, and it could be a mechanism for toxicity, but we really don't know if it is because it is incorporated into proteins, whether this is that directly affects the cells.
Okay. Yes, but it seems that there is some toxicity because the growth curves demonstrate that there is a decrease of the grow when you have cultured the cells with the increasing concentrations of selenium. Um, and about the membrane permeability, because you also mentioned that there was a modification of the profiles of the fatty acids. Did you find any any further informations which can give you informations about this, or not too much at yet?
You mean any extensive details, how it acts on the membranes and converting them to less fluids?
Yes, because the membrane is the first barrier that
Yeah.
the toxic, and it is really important that it maintains its integrity. Without its integrity, the cells dies. And you also have shown very good photos about this. So did you connect this? Not yet, probably not yet. I don't
This
know.
is work in progress.
Or
We would also say would like to
it's
see
not.
look at
Or,
this subject further. But no, the first indications that we have is that we have the techniques to look at some lipids and see that they are significantly being affected too much as opposed to the control,
mhm.
and also the photographs. But I cannot provide you with any more extensive details on the matter.
Yes, the work is great. You have a
Yeah,
lot of results and very good results.
thank you,
So I
thank
don't
you.
know if there is any further questions from the audience.
I'm
So
afraid that we need to proceed because
yes,
the time pushes us to do that. Of course, we can continue after the session any discussion that is desirable to be continued. But for now, I'm afraid that we need to continue without any more questions. And the next talk is going to be delivered by Mr. Lab, uh, and it is about the CO2 uptake enhances chromate release from fresh chromium or processing residues. Mr. Lab, the floor is yours.
Thank you for the introduction. Good morning to everyone. Just a second. I think it's... can you see the presentation?
Yeah. Yes, we can see it.
Excuse me? Okay, can you hear me?
yes.
Okay, perfect.
Yes, we can hear you clearly.
Perfect. Then good morning to everyone. My name is Florian Lapp, and I'm going to show you our results on the carbonation and the resulting chromate release from young COPR. COPR is short for chromium ore processing residues, and it's a hazardous chromate-containing waste. For those unfamiliar with COPR, it's basically the residue of the production of trivalent chromium salts, and those are used in the leather industry for leather tanning. And COPR still contains massive amounts of residual hexavalent chromium, which makes it an environmental hazard. And fresh COPR is strongly alkaline with pH values around 12, sometimes even higher. And that means that the hexavalent chromium is present as chromate. And this chromate is highly mobile. And when COPR is deposited, the chromate is leached over time. And this causes massive groundwater contamination. And amongst others, our research group has done a field investigation to the Kanpur region in the state of Uttar Pradesh in India. And India to investigate this issue of groundwater contamination. India is, I think, the world's second largest supplier and exporter of leather apparel. And the Kanpur region is a leather production hotspot in India. And Kanpur also produces its own trivalent chromium leather tanning salts. So there's a lot of COPR in Kanpur. And in this region, the COPR is often deposited at illegal and uncontrolled dump sites. And on this image on the left side, we can see one of these dump sites. First off, we can see that the material is visibly contaminated with hexavalent chromium, which can be seen on these yellow incrustations. And we can also see a gray weathering layer on the surface. And on the right side, we can see the dramatic consequence of these illegal dump sites. This is a clearly chromate contaminated groundwater well. in a rural community in Kanpur. And people living there actually use this water for their daily lives, which is yeah, quite a dramatic state in my opinion. So in order to understand the chromate leaching from CuPr, it's important to have a basic idea of its mineralogy. So here I'm giving you a brief overview of its mineralogic composition. It starts with chromium ore, which can be either ferro or magnesiochromite, which is crushed and roasted at high temperature with addition of sodium carbonate and quicklime. And this is called the high lime process. And its aim is basically to release the trivalent chromium, which is insoluble, and oxidize it to soluble hexavalent chromium, which can then be salvaged. And figure 2 here shows the concept of the process. So we can see that the calcium ions penetrate the chromite grains, which allows the trivalent chromium to escape to the outside, where it is oxidized and reacts with the sodium carbonate to form sodium chromate, which is then leached from the material in a subsequent step. And what remains after this is CuPr, which consists of relics of unreacted chromite, a mineral called brown millerite, periclase, unleached chromate, because even though it is leached, a lot of it remains, and amorphous mineral phases. So when this CuPr material is deposited, it weathers, and this happens in 2 steps. The first of which is hydration, especially of these 3 phases. And this leads to typical cement phases like portlandite, hydrogarnet, or layered double hydroxides, also called LDH. And these LDH are of particular interest because they can hold chromate in the interlayer and potentially exchange and release it. And what happens then is the carbonation of the hydrated material. The CO2 from the atmosphere is absorbed by the pore solution of the material. This leads to formation of carbonic acid, and this leads to the precipitation of calcite, to dissolution of some mineral phases, and possibly to anion exchange. And while hydration is pretty well understood and much has been published on it, there hasn't been particular research on the carbonation of CuPr. So that's what we focused on. And our main aim was to investigate the CO2 uptake capacities of CuPr and see how it feeds back on the mineralogy and the leachability of chromate. And our main hypothesis was that CO2 uptake actually enhances chromate leaching from the material. And our material were 2 fresh CuPr samples, which we retrieved directly from leather tanning salt production sites in Unnao and Vinki. And I will abbreviate these samples as F1 and F2. And here we can see a quick overview of the experiments we conducted. We performed an accelerated carbonation test in a static batch reactor for 48 hours at ambient pressure and temperature, and with 100 volume% of CO2. And then we prepared batch leachates of the carbonated and the native material to judge changes in pH and chromate leachability. Then we performed endpoint titrations with nitric acid to test the pH dependency of chromate leaching. And we also performed batch leachates with addition of excess sodium carbonate up to 1 mole per liter to test anion exchange reactions. And we prepared all of the aqueous batch leachates with a liquid to solid ratio of 10 and 24 hours agitation time. And all of the tests were performed with the CuPr's original grain size. And we performed the quantification of hexavalent chromium with direct photometry at 374 nanometers. So let's jump straight into the results. Here we can see the cumulated CO2 uptake of both samples over the whole duration of the experiment. And we can see that both samples consumed high amounts of CO2, and they showed fast kinetics. Of the total CO2 uptake, over 50% were already consumed within the first 2 hours. And the total uptake rates were 4.4 weight% CO2 for sample F1 and 5.6 weight% CO2 for sample F2. And from this carbonation, 2 main observations can be made. First, on the left side, we can see that carbonation induced a pH drop. So the native samples, which can be seen in violet, started out as strongly alkaline with a pH of 12, and carbonation reduced the pH down to around 10. This effect was weaker for sample F2, even though sample F2 consumed more CO2 and this is due to its higher acid neutralization capacity, which we determined experimentally. And on the right side, we can see that CO2 uptake caused a massive and significant increase in chromate release. The effect was especially pronounced for sample F2, where the increase in chromate release was almost 300%. And with this, our main hypothesis is basically confirmed. CO2 uptake does enhance chromate release from COPR. So next, in order to check if this was purely driven by the pH drop, so if it was a pH effect or if there was influence of the dissolved carbonate anions, we performed the endpoint titrations. And here we can see the results. The open symbols represent the pH titration steps. And we can see that for both samples, chromate release was pH dependent. Up to a pH of 9, chromate release increases, which is caused by the dissolution of chromate-bearing phases, such as chromate hydrocalamide or chromate hydrogamide. And then from pH 9 downwards, the release decreases, which is likely due to the adsorption of chromate onto freshly precipitated iron or aluminum hydroxides. But we can also see, when looking at the bold points, which represent carbonation, that titrating the samples to the same pH that was caused by carbonation released significantly less chromate. So if we assume that the chromate release caused by CO2 uptake was a pure pH effect, then the release caused by titration should be the same as the one caused by carbonation. But when we compare the bold spots with the open spots for pH 10, we can see that the release caused by carbonation was significantly higher. And this means that there must be an influence of dissolved carbonate, of the carbonate onions. And to test this, we performed the onion exchange leaching experiment. Here we can see the increasing chromate release over the range of added sodium carbonate. And we can see that indeed, dissolved carbonate released remarkable amounts of chromate with the max releases at 1 mole per liter sodium carbonate addition of over 10 grams per kilogram chromate release. And this effect is most likely due to anion exchange in these layered double hydroxides. We know from literature that, that carbonate is favorably intercalated into the interlayer of hydrotalkite, which is a magnesium-aluminium layered double hydroxide. And we also know that hydrocalamide, which is a calcium-aluminium layered double hydroxide, dissolves in the presence of carbonate. And both of these layered double hydroxides are known to carry exchangeable chromate in their interlayer. And we could also show that hydrocalamide is present in both of our samples by scanning electron microscopy, X-ray diffraction, and also thermogravimetry. But due to time constraints, we sadly can't show these mineralogic data. So to sum up the results, here in figure 8, we can see the chromate releases that were caused by the different leaching methods. And we could show that carbonation and CO2 uptake enhance chromate release from COPR. And we could also show that the addition of excess sodium carbonate is swiftly released over 50% of the total hexavalent chromium held in the samples. And as already mentioned, our data suggests that this is due to carbonate-chromate anion exchange in layered double hydroxides. So we can conclude that prolonged deposition times and thus weathering and carbonation do increase the risk of groundwater contamination at COPR dump sites. And we can also see that integrating additional steps into the production of the leather tanning salts, such as addition of sodium carbonate or carbonation, might be viable methods to reduce the environmental contamination by COPR and increase the yield for the producers, because chromate which is leached at the production site is chromate that cannot be leached at the dump site. And that's basically a win-win situation. Yes, and with this, my presentation concludes. Thanks for your attention, and I'd love to answer your questions if you have any.
Thank you very much for the excellent presentation, and thank you for being on time. It is very, very interesting, your subject, and we still have a couple of minutes for 2 quick questions, if there are any from the audience.
Okay.
Is there anybody who wants to ask any question to Mr. Lap? I would like to ask a couple of questions very, very fast. First of all, I want to congratulate you on the presentation and on the excellent picture you have for background. You probably want to tell me where you found it so I could use it myself.
What
Excellent.
figure do you mean? What figure did you mean?
The background thing that you have, the Acropolis or whatever that is.
It's that you mean, okay.
Very nice.
Thanks.
Now going to the subject, do you have any idea, because what you showed us as water coming out from a pump from a well in India, it was terrifying.
Yes.
Do you have an idea where this water is used on a daily basis?
I think it's not used for consumption, but for hygienic
I hope
purposes.
not.
But for hygienic purposes, for washing, and yeah, so people actually use this water, but I think not for consumption. And I think there had been plans to implement a study on a bucket solution where people could reduce the hexavalent chromium to trivalent chromium in their homes. But then COVID happened, and I think the study was never conducted. So now there are plans to remove the COPR dump site, but it's an ongoing issue.
It's an amazing. Well, the leather production in India is always, as you said, a very important industry, and all the consequences of that industry and in the environment and to the people is tremendous. But anyway, I want to ask you that you measured chromium spectrophotometrically on the 374 nanometers, you said?
Indeed.
Okay. Do you know if there are any interferences by other species in that wavelength?
I think there are not. The point is that the main ion in our samples is chromate. All the other bulk anions, I think, are present in much lower quantities. But there are no interferences that I know of.
Did you have the opportunity to check that with another technique like ICP-MS or something like that?
We did not. In the beginning, we used the more traditional method at 550 nanometer, the DPC method, where we basically reduce the hexavalent to trivalent chromium, which creates a purple complex, which is then measured. But the disadvantage of that method is that it has a much lower calibration range. I think it's from 0 to 1 milligrams per liter. And we have chromate concentrations of sometimes up to 1,600 milligrams per liter. And the method we used has a calibration range of 1 to 25 milligrams per liter. So by using it, we could go for dilutions that were not as extreme, which reduced our measurement errors. And also the direct method has less prior preparation, which allowed us to an easier measurement of multiple samples.
Okay. And finally, you say that bicarbonation of the material could reduce the environmental risk. Am I right on that conclusion you made?
Yes.
Okay. How feasible that is to be done on a practical basis?
Yeah, that's a good question. So there have been experiments on accelerated carbonation in reactors that attern, and in theory, I mean, it's a roasting process. So there are CO2 fumes, and those could be used in theory to carbonate the material. I think in theory it could be feasible, but it has not been implemented yet. So
very difficult. Thank you very, very much again for your excellent presentation. And Professor Soares is going to present the next speaker. Helena, your micro.
Sorry. So the next speaker will be Soltanidis. and the title of the presentation is The Effect of pH on the Release of Metals from Conventional and Coal Cement Blocks. Please, Soltanidis, can you start? The floor is yours.
Thank you, Professor Soares. Just a moment, please. Come on. Can you all see my screen now?
Yes.
My camera is not a good. Is it better now? On my head. Okay, I have a laptop, but nearest to the camera. Okay. Now it's
Much
better.
better.
Much better. Okay.
Much better.
Good morning, everyone.
Yes, better.
Excuse me?
Go on, please. Sorry.
Okay. My name is Vangelis Sultanidis. I'm a PhD candidate in the Department of Environmental Engineering, Democritus University of Thrace. And the title of my presentation is The Effect of pH on Release of Metals from Conventional and Cool Cement Blocks. First, a small introduction. Generally, cement blocks are used for constructions of roads, sidewalks, parking areas, and squares, which occupy a significant fraction from 29 to 44% of a contemporary urban area. Pavements constructed from conventional cement blocks absorb solar radiation during the day and release into the atmosphere at night. In contrast, cool pavements reflect a fraction of solar radiation back to the sky, and this results in lower surface temperature and mitigation of the heat island effect. Cool materials do not absorb large amounts of sunlight and do not store much heat in their mass. They are characterized by high solar reflectance and high infrared emittance. There are cool materials for buildings like coatings and membranes, and cool materials for urban surfaces like cool asphalt, sidewalk tiles, and cement blocks. Now, the cement blocks are compact prefabricated concrete elements in various shapes and dimensions with a minimum height of 6 centimeters and a wide variety of colors. All paver blocks must comply with the Greek standard EN 1338 of 2003. Some common applications of this are for the construction of pavements, yards, squares, and streets. Now, for the problem definition, today the waste volume of cool materials is relatively small, but the wear that will suffer over time will lead to their gradual replacement and disposal. Conventional cool barrier pavements in contact with rainwater will produce leachates during their service life, reuse, recycling, or disposal, which may affect soil, surface water, and groundwater quality. One way to judge the environmental impact of conventional cool cement blocks is to assess their potential release of chemical constituents by leaching characterization. So the objective of this work was to determine and compare the leaching behavior of heavy metals from conventional cool cement blocks as a function of pH. These two photos show cool and conventional cement blocks. Excuse me. I have a pointer. The cement blocks that we used in our study, the blocks were purchased from a Greek manufacturing company of cool and conventional construction materials, and both of them had the same shape, size, and color. We thought that the red top color section of the cool block had some ingredients that make it different from the conventional block. So the first step was to separate them in two parts, the red top section and the gray bottom section. So there were 4 different materials to be tested, the cool top, the cool bottom, conventional top, and the conventional bottom section. And finally, we granulated them for the tests. For the determination of the total environmentally available metal content, we use method 3050B from the US EPA. It's a strong acid digestion of sediments, sludge, and soils. The effect of pH on heavy metal leaching was determined using the batch leaching test. It's the method CNPS 14429. This method needed more than 95% of the block's mass fraction having a particle size less than 1 millimeter. And last was the tank leaching test, which was used to determine the rate of release from the blocks according to method 1315. This method uses monolithic materials, so the test was applied on intact section of the blocks of known geometry, fully immersed in a deionized water tank with pre-scheduled renewal of the leaching solution. And finally, the leach excess light was used as the data management tool. All samples were analyzed by flame atomic absorption spectrophotometry. The selected metals were cadmium, cobalt, chromium, iron, manganese, nickel, lead, and zinc. And also black experiments were conducted to check the quality control of the measurements. And let's start with the results. First, the moisture content and natural pH is in this table. Cool top cement blocks have higher natural pH compared to cool bottom and conventional top. Also, the total heavy metal content as determined by acid digestion. Cool top blocks have higher lead, zinc contents than and lower chromium. Chromium is here. Chromium, iron, and manganese contents compared to conventional tops, and they also have higher lead and zinc contents and lower iron and manganese contents compared to cool bottoms. These figures show the comparison of the pH leaching from cool and conventional blocks from nickel, lead, and cadmium. The straight lines here show the waste landfill limits according to the European legislation. It appears that the leaching curves of these figures are very consistent and close to each other, indicating that the leaching mechanisms are the same for all cement blocks. Some limits for inert waste landfill are exceeded, but I forgot to cycle the natural pH samples. But as we saw previously, it was around 12.5. And all the results showed that leaching was higher at low pH and decreased with increasing pH for all metals. The next figure shows the pH leaching for copper, zinc, and chromium. Also, the leaching curves of these figures are very consistent and close to each other, except chromium, where we have different release curves. Also, chromium exceeded the limits for inert waste landfill. And the same leaching curves for manganese, cobalt, and iron. Here we have an example of typical leaching data for manganese according to method 1315. The data include the variation of leached pH, leached metal concentration, cumulative metal release, and flux, all as a function of cumulative leaching time, which was 63 days according to the method. The pH variation with time was common for all heavy metals studied. In the first figure, pH curves of all blocks had a range from 8.2 to 10.1, with the bottom section of the cool block exhibiting the highest pH values. They all follow an increasing trend for the first 2 days, and then they decrease, indicating depletion of the water-soluble alkaline constituents. In the second figure, leachate concentrations for manganese are below 0.1 milligram per liter and do not show significant variation of the leaching time. Data from the top-bottom section of the cool blocks overlap, while bottom section leachate concentration are higher than those from the top section of the conventional blocks. This order in profiles is also observed in the third and fourth figures. And here, if the leaching process is diffusion controlled, the cumulative release is expected to be proportional to square root of time, and the flux proportional to the inverse square root of time. So the straight lines here with slope equal to 0.5 and negative 0.5 for the cumulative release and the flux correspond to diffusion control leaching. Sections of these lines with slope other than 0.5 indicate a different leaching mechanism. These figures show the effect of pH on nickel, cadmium, and lead leaching together with mass transfer data from the tank leaching test. All data from the tank leaching test for nickel, cadmium, and most of lead are shown below the leaching curves. This indicates below the leaching test, except of lead, where some samples was not reached during the mass test. The concentration, some lead concentration of the tank test appear to approach the equilibrium. This is most likely due to dissolution of surface minerals rather than mass transfer through the cement blocks. Now, for the determination of the diffusion coefficients, we use fixed second law analytical solution. And for that, we needed to calculate the cumulative mass release, density, maximum leachable quantity, and the leaching time. The observed diffusion coefficients are presented in this table. Some values differ by several orders of magnitude. For example, the diffusion coefficient of manganese 1.6 times 10 to the minus 15, and from the conventional top section is 7 times 10 to the minus 20. Also, the diffusion coefficient of lead is 6 times 10 to the minus 16. From the conventional top is 3 times 10 to the minus 13, and for the conventional bottom, 2.2 times 10 to the minus 11. In the empty cells, the release is not controlled by diffusion, but from other mechanisms. For example, it could be the initial surface wash-off. The determination of diffusion coefficients now can help us to predict the release of constituents in the environment and also to evaluate different management scenarios. For example, in these figures, we can predict the soil concentration of cobalt and iron after 10 years of release. And finally, the conclusions. Results from batch experiments showed that leaching was higher at low pH and decreased with increasing pH for all metals. Since some limits for inert waste landfill are exceeded, disposal of both cool and conventional cement blocks to non-hazardous waste landfills is permitted. Leaching curves as function of pH are very consistent and close to each other, indicating that the leaching mechanisms are the same for all cement blocks. The impact of cool materials is not different or not worse than the conventional ones. And observed diffusivity values differ by several orders of magnitude and are very low, indicating very slow diffusion. And thank you very much for your attention.
Thank you, Sultanadis, for your nice presentation. Okay. As I can see from the link of the questions, it seems that there are no questions related directly with your presentation. I don't know if anybody from the audience want to do any questions. Is there any question? No?
I'm having
so I have a question. Sorry?
I'm having a little remark on the live stream, because I think in the live stream there is this bar on the downside, which I think in the live stream covers part of the presentation. Maybe that can be fixed for the next presentations.
You couldn't see my the entire presentation? You had a problem?
No, I think it's that in the live stream, the presentation is equipped with this bar, which shows the title of the presentation at the bottom. And this bar covers some part of the presentation. I think this was the case for all presentations so far.
Well, I'm not sure about it, but probably the support team is listening to your comment, and they will fix it probably. Is the support team with us? Στο live stream φαίνεται μία μπάρα που εμποδίζει όλη την παρουσίαση, να φαίνεται όλη η παρουσίαση. Αν θέλετε να το τσεκάρετε.
Αυτό που φαινόταν κάτω ήταν, είναι μάλλον.
Okay. So the support team is going to take care of that comment, and we thank you very much for doing it. Most likely, we do not have any free time to continue with this presentation, and at this point, I would like to ask if we have any presenters for the Flash poster presentations. Starting with Dr. Kelepedzis. Is Dr. Kelepedzis or anyone from his group with us? I guess not. The next.
I think I
part. Okay, so there's a video. Yeah, there is a video for that Flash presentation, and I guess the supporting team is going to upload it, right? Θα το ανεβάσετε εσείς? Okay.
Hi to all. The title of my presentation is Coupling Lead and Zinc Bioaccessibility with Sequential and Nitric Acid Extraction in Soil from the Industrial Area of Volos. I want to give you some background information. So, risk assessment based on total concentration of elements may overestimate the actual risks for the environment, including human beings. For this reason, single and sequential extraction procedures are applied in order to determine the mobility, the activity, geochemical fractionation, and human oral bioaccessibility of metals. It is noted that the oral bioaccessibility is the fraction of the element that is soluble in the gastrointestinal environment. So the principal aim of this study is to couple lead and zinc bioaccessibility with sequential and nitric acid extraction in the industrial area of Volos. We collected 29 soil samples around the Velestino Steel Factory, within the urban area of Volos, and around the cement plant at the east part of the city. In these soil samples, we determined the total content of elements by a strong acid dissolution, the reactive fraction, the bioaccessible fraction, and also we performed the PCR sequential extraction procedure, which separates the elements into 4 fractions: the acid soluble, the reducible, the oxidizable, and the residual. Moving to the results, as you can see this figure, we have similar bioaccessible amounts for lead and zinc. Apparently, we have higher reactivity for lead compared to zinc. Lead is mostly bound to iron and manganese oxides, whereas the geochemical partitioning of zinc is dominated by the residual fraction. Interestingly, we found very high correlation coefficients between the bioaccessibility of metals and the sum of the first three fractions of the BCR, which is the mobile fraction, the green color in this figure. And also, we found very high correlation coefficients between the bioaccessibility of metals and the reactivity of elements, the red color in this figure. As conclusions, we can say that lead and zinc bioaccessibility is governed by the mobile pools of lead and zinc present in the soil, and also we can use the simple dilute nitric acid extraction in order to determine the bioaccessibility of metals in urban soil influenced by industrial emissions. I would like to thank you for your attention, and also for more details, you can read our very recent publications in Science of the Total Environment journal.
Nikos, we cannot hear you. Your microphone is...
I'm sorry, I guess now you can hear me.
Yes.
Probably we can proceed to the second flash presentation, if there is a video available.
Good morning, everyone. My name is Antifanti and I'm a PhD candidate. Today, I would like to present the results from the first attempt to evaluate mercury levels in the coastal waters of Greece in the last 8 years and identify trends and spatial patterns. Some basic facts about mercury are the recognized toxicity to humans and wildlife, several natural and anthropogenic sources, a complex biogeochemical cycle, increased lability, re-emission to the atmosphere, and the known bioaccumulation and biomagnification along trophic webs. The study of mercury is of great importance for the Mediterranean region due to its increased abundance in the geological background. Mercury is a priority pollutant in European Union environmental legislation and international initiatives. Water sampling was conducted during 8 years of monitoring. The 2017 campaign took place only in Salonikos Gulf, and the stations were depicted are depicted in this picture. In this figure, you can see WFT network covering the major coastal areas and gulfs of Greece. In total, more than 500 water samples were collected with the Hellenic Center for Marine Research, Oceanographic Basins, Aegean Philia, and analyzed for total mercury using VPA 1631 method. The mercury data were subject to exploratory statistical analysis. Now let's move to the results. The median mercury concentrations of the entire data set was calculated at 2.2 nanogram per liter. This table presents the number of samples, the percent of values above 4 nanogram per liter, mean, median, and range of total mercury expressed in the nanogram per liter. I would like to bring your attention to this chart. The area which can be identified as a hotspot is Elefsina Bay. In all other areas, the median values were close to the dataset, a median of 2.2 nanogram per liter, as would be expected. In Inner Saronikos, the increased mercury concentrations are associated with the wastewater treatment plant outfall and atmospheric contributions of the metropolitan city of Athens. I would like to summarize by saying that the mercury levels measured in all coastal areas of Greece were below the European legislation threshold of 70 nanogram per liter. It becomes apparent that stations near ports, major cities, motorways, river estuaries, volcanic environments, and mining activities present some of the highest values. The mercury level of coastal waters of Greece and the mercury levels of deep water of Aegean and the Ionian Sea should be correlated with mercury levels in fish and seafood, since there are some indications of elevated mercury levels in edible marine biota. Thank you for your interest and your attention.
Very good. Let's now go to the next. Very good. let's start with it.
Heavy metals in soil, sediments, and water from Lake Ohrid catchment in Albania. I've worked on this project with my colleagues from the Agricultural University of Tirana, and it's also been supported by CEST, so the Critical Ecosystem Partnership Fund. The study focused on investigating ultramafic regions where dump sites or mining activities could have impacted the aquatic environment in lakes. We focused on Lake Ohrid, one of the oldest in Europe, shared between Albania and Macedonia. And the investigation focused on seeing if sources of heavy metal, mostly nickel, chromium, and cobalt, were responsible for any contamination on the water of Lake Ohrid.
Hello everyone, my name is Austin. I would like to shortly present you the results of my research on changes of photosynthetic performance of brassica nipas affecting cadmium phytoextraction performance under elevated temperature, the concentration of carbon dioxide, and drought. Recent studies have shown that under higher temperature and elevated concentration of carbon dioxide, the photosynthetic potential and biomass production of reefs increase significantly. That can improve the potential for cadmium phytoextraction by reefs in the future. Therefore, the aim of the study was to evaluate the cadmium removal efficiency from the soil by reefs under changing climate conditions. At the elevated temperature and concentration of carbon dioxide with or without drought stress, using chlorophyll efflorescence parameters as photosynthetic performance indicators affecting cadmium phytoextraction performance. The experiment was conducted in the growth chambers under a controlled environment. In half of the pots, the soil was packed with cadmium chloride to get a cadmium concentration of 100 milligrams per kilogram. Then half of the pots were placed in a phytochamber with the ambient temperature and carbon dioxide conditions, and the other half of the pots were placed in the phytochamber with elevated temperature and carbon dioxide conditions. Drought stress plants were maintained under 5% of volumetric soil water content, while control plants were kept under 30% of volumetric soil water content. The duration of cadmium treatment under both climatic conditions was 43 days, and the duration of drought stress under elevated temperature carbon dioxide conditions was 17 days. Here you can see the results, and the main findings of the study are the facts that with the less affected photochemical quantum yields and better general physiological state of photosystem 2, grapes grow under elevated temperature and carbon dioxide conditions, produced significantly higher harvestable biomass, and extracted significantly higher cadmium content from the soil than those grow under ambient temperature and carbon dioxide conditions. Drought diminished the positive elevated temperature and carbon dioxide impact on cadmium phytoextraction significantly, and the difference from the control plants under elevated temperature and carbon dioxide conditions with drought stress was insignificant. However, drought stress reefs grow under elevated temperature and carbon dioxide conditions, demonstrated unchanged photochemical activity due to suppression of cadmium, suggesting other restrictions to photosynthesis than the decrease in electron transport flow. Thank you for your attention, and I hope to see you during the poster session. See you later.
Okay, and there is one more presentation left. Let's go for it.
Heavy metals in soil, sediments, and water from Lake Ohrid catchment in Albania. I've worked on this project with my colleagues from the Agricultural University of Tirana, and it's also been supported by CEST, so the Critical Ecosystem Partnership Fund. The study focused on investigating ultramafic regions where dump sites or mining activities could have impacted the aquatic environment in lakes. We focused on Lake Ohrid, one of the oldest in Europe, shared between Albania and Macedonia. And the investigation focused on seeing if sources of heavy metal, mostly nickel, chromium, and cobalt, were responsible for any contamination on the water of Lake Ohrid. The material and methods used, we focused on collecting water samples from soil, water, and sediments in different areas. Specifically, the metal in the solutions were determined directly by the graphite furnace AAS, and we used a measuring of a multi-parameter portable instrument to determine the samples. From the results, we discovered that the physical and chemical characteristics of the water temperature were acceptable within the limits of survival and metabolism, as well as the same could be said for the pH values. However, when we investigated the heavy metals in the soil, we discovered that around the ultramafic regions, we found high levels of nickel, chromium, cobalt, and iron. The highest metals obviously were found around these areas with mining activities in ultramafic region. The same could be said for the metals, heavy metals in the sediments collected around the lakes. These were very high in the areas where ultramafic regions were focused, such as Piscopat, Poisk, and Hudanisht. When we investigated the distribution of heavy metals in the water, however, we found that in the surface of Lake Ori, there was no clear presence of all of these heavy metals except for iron, which was quite high compared to the normal levels, as you can see in the table attached. As a conclusion, the study focused on assessing the risk on the environment concerning these heavy metals on these ultramafic regions, and discovered that the basic environmental quality standards for the surface water were acceptable except for iron. Thank you.
Okay, thank you very much. This was the final flash presentation. Professor Soares, I would like you to do a very short closing for this session, if you don't mind.
Okay, so I would like to thank together with Professor Nikos for all of you for this nice session, for all your contributions, which were in heavy metals, but in different ways, in different perspectives. So it was a pleasure from our side to be with all of you today. We wish you all a good, a nice and conference and all the best for the future. And thank you for being with us.
From my side, thank you all for being here. Thank you, Professor Soares, for sharing the chairing of this session with me. And I hope that you all enjoy the conference, and you all will be, since you are all young scientists, you will be productive with your future work, and you will enjoy science for the many years to come, all of you. Thank you again, and we need to close the session now, so enjoy the rest of the conference. Thank you very much. Μπορούμε να κλείσουμε τώρα.
Thank you.
είτε από chat είτε από οπουδήποτε. Οκ, κανένα πρόβλημα. Έγινε οκ. Ναι, είναι 12 λεπτά και 3 λεπτά για Q&A. Αυτό λέω, ότι πρέπει να αλλάζουμε link για αυτά τα 3 λεπτά. Δεν νομίζω, δεν είναι. Ναι, οκ. Ναι. Για να δούμε. Εγώ κανένα πρόβλημα. Ναι. Ναι. Μισό λεπτό δώστε μου και ξεκινάμε. Μισό λεπτό.
So good morning, everybody.
Hello. Hello. Okay, I would like to welcome everybody to session 12. Okay.
Ο κύριος Δερματός παρουσιάζει, εγώ θα πω δύο λόγια, αλλά και ο κύριος Δερματός παρουσιάζει την προσκεκλημένη. Ναι.
I would like to welcome everybody on session 12, soil and groundwater contamination and remediation, under the auspices of CEST 2021. And at this point, I would like to introduce our keynote speaker, Margot de Kleen. Margot is a senior soil and water advisor in the Dutch Ministry of Infrastructure and Water Management. She is a member of the expert group Soils of the European Commission and the Common Forum Network. As an advisor on soil and water policy at the Dutch Ministry of Infrastructure and Water Management, she promotes sustainable use of land and soil as essential for achieving the UN Sustainable Development Goals. Mrs. de Kleen is going to discuss the key parameter of collaboration for a successful policy in the Netherlands, The exact title is Soil Policy in the Netherlands, Collaboration is the Key to Impact. And at this point, Margot, I would like to extend our special thanks for your presentation here today, as well for your presentation tomorrow on the workshop on contaminated soil management in Greece, learning from EU and international experience, which is going to be an event, the tomorrow's event, an all-day event, and with more than 300 participants at this point from all around the world on a very, very, very interesting and complicated, believe me, subject. Margot, you have 30 minutes for your keynote presentation, and I will give you the floor right now. Thank you.
Thank you very much. Thank you very much, Mr. Dimitris Dermatas. I'm really honored to be able to give a lecture here on the work we've been doing in the Netherlands for the last time. Sorry for the leap of time because I'm still in the Netherlands. It's 11:37 now, and I was expecting to give this presentation 1 hour later, but you are ahead of us in time. So, well, I hope this lecture will be interesting for you, and I hope I'll be able to share my screen with you, and otherwise I would ask you to share the presentation I sent earlier to you. I have a small problem here. Sorry,
You're trying to share your screen, right?
Yes. So
it's in the bottom. I was not also familiar with. Yeah.
I think it will be possible to share my screen.
Exactly.
Now, can you see my screen? Not yet. I understand.
If I may, okay.
You can see my screen now?
Yes, beautifully. Margot, everything looks fine. Perfect.
Oh, so now, not so perfect as I hoped it was. I'm really sorry for this.
No problem, Margot. Don't worry about it.
That's fine now.
That's fine
now.
You
can see my whole screen because I don't succeed in putting it in the presentation mode. Yes, now I did.
Yes. Perfect.
So I'll try to get rid of this. So, sorry. Well, it's all about policy making, soil policy making, what we have done in the Netherlands for already quite some time. And I want to take you into the process we went through in the Netherlands, where we found out that collaboration in soil policy making and policy making actually as a whole is really the key to have impact. And I also want to state that this is not something you do easily. It asks a lot of changes in mindset and also in looking upon policy making as a whole. Like you actually need a paradigm shift. The way we behave towards our soils is actually that we behave it like slave drivers. We take everything out of our soil and leave it behind in a state of contamination, like it's barren, no more organic carbon, they are dry, they are bare. And this could be so much other. And what you see often also in policy making is that from such a behavior comes a kind of act that we want to protect our soils. We want to behave like a mother towards our soils. But then also you don't let the soil flower and use all its functions in the way it could give you profit and also profit itself. So what we actually aim for now is to see soil as our partner in which you invest. And if you invest in soils, it also invests in you because it gives you welfare and it takes care for your well-being as people, but also as your planet. So if I want to talk about policy making in the Netherlands, I thought maybe I should just start with all the lessons we learned over the last decades, actually. And what's very important for policy making is actually that you have to be assured of political commitment. It all starts with political commitment. And then if you have this commitment, what's also very important is to stop your behavior in which you are degrading the soil. So start with prevention when you are talking about contamination. Prevention is really the starting point of soil policy. Also important when you want to make up your policy is that you know your problem. Where are you talking about? What's the area? Where are the areas which are contaminated or degraded and what caused them? How important is this contamination? Is it stable or is it moving? Are there acute risks or not? So it's key that you find out the problem and how big it is. It's also important to realize that if you set up policy, if you make very strict laws, you can have endless juridical procedures. They are long and you cannot get into action and actually come to the core of what you aim for, which is just clean up and manage your soil sustainably. So try also within your policy to find ways to avoid long and costly juridical procedures. And well, it's always a fact that authorities need to take the lead. They have to set the goals and the ambitions and also the framework. As important is that you don't have to have discussions about risk, what is a risk or whatever. So you need to prevent juridification of risk by communicating clearly what you define as risk. So you can have good communication with public and other stakeholders using the soil. Then part of your policy making, of course, is that you have to organize it, because you can make a law, but people cannot act and come into action if you don't have organized form of quality control and quality assurance. And if you don't have build up capacity for the organizations, for your other authorities. What we learned in the Netherlands is that it's not only a thing to be done by authorities, and that if you want to speed up the execution of soil remediation, covenants and branch programs are really very helpful. But if you want to draw up such covenants, then you need trust one another and you need common language together. And what's also very helpful, especially when you don't have a lot of funding, to combine remediation and management with spatial developments. When you go and make your policy, it's also very important to realize in what kind of surroundings you are living, because policies are actually really determined by cultural, social, economic, and natural characteristics. So what works for the Netherlands does not immediately work for Greece, for example. If you look at the inhabitants in Greece and in the Netherlands, you see that in the Netherlands we have quite some more people living than you have in Greece. And if you look at the surface, you also see that we have more than 400 persons living per square kilometer, while you only have 80. So this means kind of thing in the pressure on land, but also possibly on the activities which have taken place in our countries and the amount of contaminated places we found. Also, it's important to know what are your challenges. And when we look into the Netherlands, we see that what we want to do on our surface is quite a lot. Our main challenges, possibly also in Greece, are climate change, but also a change in energy. We are moving towards a sustainable energy, and it asks also a lot of distribution. We want to live in healthy cities, and we need a lot more housing for all our inhabitants. We want to have sustainable mobility. We want sustainable food production, and we are aiming for a circular economy. And what all these challenges have in common is that they need land, and they need clean land. So they need clean soils. So those challenges are also connected to how our soil policy should be given form. So reasons for soil policy, well, in the Netherlands, it is the intensive use of land. Also that we have a lot of water and groundwater, and we use this groundwater for drinking water. We started with 400,000 potentially polluted sites in 2010. And well, we have been able to bring this amount down to known sites, and all those known sites with actual risks are now under control. But we still have areas also with diffuse pollution. And soil policy is important because the economic value of land is so high, and especially of clean land, of course. And our groundwater, which is still very precious, but also under stress of contamination, is near to the top layer of the soil. As I already said, soils are essential for our welfare and well-being. The soil functions, or the ecosystem services, they help us with crop production. They stock our drinking water and groundwater, but we also need them for as a carbon sink, as water storage, and for stability. So all these functions are essential, and they are also a kind of a clue and way to solving our main sustainable development goals, our big societal challenges. And what we see is that soil quality is under pressure, not only by contamination, but also by degradation in other forms. Maybe in your country it's soil erosion, but in the Netherlands it's also drought. It's a loss of soil fertility, it's salinization, it's soil subsidence. So it's really important to have a good soil policy. And our soil policy actually started with soil contamination. Well, if you look at soil policy in the Netherlands, we have 35 years behind us, and we always went for a three-way approach. We started, as I said, with prevention, and we draw up well, a whole inventory and guideline how to prevent further contamination. What we also made was a pillar of management of light polluted soils, because we very soon found out that we were not able to clean our soils up to the multi-functional use. So we had to find a way how to cope with light polluted soils and draw up a framework around that. And of course, we also had a pillar or a way to remediate soils, but that we found out was best done risk-based. And we developed this concept for historical sites, and the government pays partly for historical sites contaminated before 1987. Because that's when our first law on soil remediation became effective. To start a soil policy, we did an inventory of contaminated sites. As I said, at the highest point, we had 400,000 potentially contaminated sites, which we were able to bring down to sites which were known and with actual risks, and they are now all under control. We drew up juridical instruments because, of course, we also needed legislation. We needed to make clear who is liable and when, and we also made a standard framework for values and when something is a risk and when not. But we also drew up an organization. We have competent authorities. We started with the national government as a competent authority, but later on we had also the big all the provinces and the big the big authorities from the big cities being a competent authority, because they knew best what sites were locally contaminated, and they also knew what is locally what are the local characteristics. We also had to organize laboratories and investigation protocols, because we want to have a clear and certified way of how you do your research and how you do your monitoring. And what was also important is to know what kind of costs are we looking at, how expensive are those remediations, and what kind of reservations do we need to make, and do we need others to ask to make for cleaning their plots. Coming together with that is also that, of course, knowledge and technology development were important, especially in the first years, because then nothing was known. And we found out that knowledge development and technology development together with our national institutes and businesses worked best. So we had big development programs and knowledge programs, and this worked out really good. We also drew up process management with quality assurance and quality control, because there's a lot of money involved and you don't want to have fraud all over the place. And to know what you have done and learn, it's also very important to monitor and to collect data and information, and also be transparent in that, and make it accessible for all stakeholders. But as I said, not only legislation leads to success. What we did also during the time was make a branch programs together with the industries for gasworks and railroads, but also for filling stations, dry cleaners, and national property like military sites, or also the works from my own organization with the infrastructure. We drew up covenants. In 2001, we had a covenant from the national authority with the industries, but the last 10 years we had a covenant between the public authorities and also industries, which worked out very well. We also introduced the area approach, because we had a lot of contaminations, especially in cities. And for example, the harbor of Rotterdam, where we had contaminated groundwater, but we couldn't indicate the exact source to the one who had inflicted this contamination. So we worked on area approach, where all the industries together pay together for the monitoring for the joint plumes. We also have in the city of Utrecht a combination of an area approach where we combine remediation together with heat-cooled storage, where we work together with energy providers and the businesses which are situated in this area. So there's a joint benefit and cost business model. And we worked also on transfer of liability, because if you want to take over, for example, the monitoring of an area with contaminated groundwater, then you have to make a transfer of liability to enforce the industries to work together. Another approach is the Green Deal. We had different Green Deals, which were not so focused on soil policy regarding contamination, but for other soil functions, like for temporary nature, farmers for water, and infra-nature. And our goals with all those kind of collaborations were that we wanted action. We want to get things done. And if you are able to draw up an agreement, you get into action. It's also leading to a shared responsibility, and often also to joint financing, because you bring stakeholders together which all have a certain interest. So together you can achieve something, and you can also join your costs, so then it becomes more feasible for everybody. Prevention also is an important goal, because if you do nothing and you stay out of action, contaminations can spread and situation can get worse and costs become bigger. Another goal is that you work on innovations, because you bring people together, which leads to a creation of new examples, and thus innovate also, like for example, the cold storage together with contamination is a kind of innovation. And what we are also working on is land stewardship. So all the services soil offers can be come to as a value for the whole community, instead of for some private owners only. So why collaboration is so important, and why is it so successful? Because with collaboration, you avoid long and costly juridical procedures. You share the costs and benefits, because you create value, and because you know who are the stakeholders and you ask them to join. So you can combine national, regional, and private interests, which is really, a good thing, and also very attractive. Also, you create business development by innovation potential, and also this is very nice export product. And you, at the same time, you develop new knowledge, which makes you understand the processes in the soil, which lead to transparency and enables quality and control. I want to share an example of value creation to underline the importance, because if the land value by reclamation is becoming really high, while the reclamation costs are not so high, you often see that private parties just go ahead. They don't need money from an authority or whatever. But when the reclamation costs are quite high and the value of land is not so high, you see that mostly the payment has to be done by the by the public, by the local authorities. And actually what you want is that as much as possible, private parties and public parties work together, so that not only the public has to pay for all the sites which are not dynamic. And how are you going to achieve that? Because that's the question. Well, one of the possibilities is that you create collaboration. So one of the things is you have to show the value of the clean land for public and private parties. And how can you do that? Just to connect with societal challenges. If you clean up this land, can it contribute to sustainable energy, for example, or to climate adaptation or mitigation? What stakeholders are involved in those processes, and connect then with them. And also sometimes it works to scale up the area, because if you go and look for opportunities in a bigger area, you find more stakeholders which have interests, and then you can share those interests, and then you can get the cost of the reclamation brought down, because there are so much people interested in the value you are creating. And collaboration, you must know, has roles and rules, because nothing is free, and collaboration, also a covenant or a Green Deal, is not free. It has a few roles. For example, authorities should take the lead. They should set a goal and ambitions and a juridical framework, but also certification, knowledge development, and data collection and monitoring are really roles authorities should take. At the same time, liable parties also have a role. They need to take the responsibility and make reservations for remediation costs now or in the future. They also need not to lay back, but get into action, and they should be open for collaboration and be transparent on data and share knowledge. Other stakeholders, like citizens or energy companies, spatial planners or developers, can also take responsibility. They can invest with knowledge and innovation and means, and they can also address the benefits. And collaboration, as I said, is not free. It's also not that you don't have to make agreements, because you need to make agreements and they need to be signed. In that sense, they have a kind of legal basis, because you need to make clear what are the tasks. You need to set a time frame and a strategy and implementation plans with goals, and also the results you want to achieve. And monitoring, evaluation, and aftercare are also part of these signed agreements. So no free riders, but you need trust in the parties with whom you are going to collaborate. This asks for a joint language, because not all the languages are the same. And then I don't mean that Greek and Dutch are different languages, but also that soil sector has a different language than the spatial planners or the energy sector. And you need to find a way how to communicate together. You have to be transparent, and you have to take care for continuity. So the dynamics of collaboration and the change over 35 years in the Netherlands can be shown by this slide. We started in the 1980s with a crisis management, and we came to policy development. From multifunctional use of soils after remediation, we went to a risk-based approach. When we started with authorities in the lead and also being the responsible, the responsibility for remediation, we changed it to the private sector who had to take the responsibility. And instead of a central authority, the authorities became decentralized. We started also with a voluntary approach, but it became an obligation. And instead of that, we were going to pick up all sites which were contaminated. We only went to sites with actual risks. Instead of looking only at the environment, we put the remediation as part of spatial planning. And instead of excavating sites, we now have more and more in situ techniques. Legislation as a starting point is becoming more and more flexible instruments together with others. And instead of risk control only, we work towards value creation. Does this mean we are ready and have everything under control? I'm afraid to say we still have a lot of challenges. We have diffuse contamination, like the whole PFAS crisis, which we share with a lot of European countries, and also emerging contaminants are still on our agenda. Groundwater management becoming more and more important. We have to suffer from illegal dump sites with waste from drugs processing, and we also have a lot of aftercare projects going on, which we want to stop by creating resilient themes. But still, what I want to share with you, that is, if you take care of soil, the soil take care of you. And we, well, that's our challenge for the coming years. And if you want to have more information about how we organized our policy, we have a booklet in English. It's called Into the Soils, and you can find it on the site of our organization. So thank you very much for your attention. And if there's still time, please, I'm open for questions.
Yes, at this point, I would like to thank you very, very much, Margot, for what was a very interesting and thorough presentation all across the domain, all across literally the domain from start to finish, and also with a very good clarification at the end that the war is still on with trying to protect soil, because, you know, the threats are different and there are still things that are unresolved. I can attest to that also from academic standpoint. Are there any questions for Margot at this point by anybody?
Dimitris, if you may, I have a question for Margot.
Yes, please.
First of all, I would like personally to thank Margot de Kleen for all her help that has given already for you know, framework in Greece that we are trying to construct these days. So my question is, how do you think, Margot, that stakeholder consultation can be improved, and in order to have valuable outcomes? And if there are stakeholder organizations active in the Netherlands right now, talking with the ministry and all the other players?
Yeah, thank you, Iraklis, for your question. Well, in the Netherlands, I think we have a lot of stakeholder groups, and we have a lot of projects going on between stakeholders and the authorities. It's very, how do I say, very broad, because we have special agreements with industries and authorities on remediation of industrial sites. So together with them, we build a knowledge center, and also a kind of, well, simple remediation procedures. So that's a form of stakeholder involvement, but we've also completely different stakeholder groups where we have a whole community of soil ambassadors which are discussing together how to improve the soil quality in the Netherlands and what kind of actions we should undertake. They drew up a kind of manifest, a Terra Agendum, and they try to get new aspects on the political agenda. And there are also all kinds of pilots going on how can we draw up a subsurface plan for our city, for example, in Utrecht and Enschede, cities in the Netherlands. There are plans for the subsurface in which all kind of actions are planned for storage of heat, cold, where can that take place, reservations for groundwater, but also reservations for pipelines and sewers and those kind of activities.
Thanks a lot.
Thank you very much, Margot, again. Is there any other question for Margot? Okay, because we started a bit late, Margot, I saved my question for tomorrow because it involves the impact of the climate crisis as we live it through right now and what your view on that is, because we're still contemplating, I believe, on a global basis, everybody's contemplating, what that game is, but I'll hold this for tomorrow. And maybe that will entice some of our other viewers at this point, members and presenters for this session, to join the contaminated soils workshop tomorrow. I just want to stress that you need to register for that. It's free online. You need to register for that. That's all. And at this point, I would like to introduce briefly myself and Dr. Panagiotakis and pass the ball to him. My name is Dimitris Dermatas, and I'm a professor at the National Technical University of Athens in the School of Civil Engineering, and I'm a professor of Geoenvironmental Engineering. And Dr. Panagiotakis is a colleague in many of the research projects, and mostly applied research projects so far. He runs his own consulting company, Enidron, and he also got his PhD. He did get his PhD from National Technical University of Athens. Iraklis, Dr. Panagiotakis, the floor is yours to introduce the next speaker.
Thanks a lot, Dimitris. The next speaker is, we have a video actually. It's about the trifluoralin polluted soil treatment using nanosecond pulse DBD plasma. And the authors are Ms. Hadisimeon, Dr. Tataraki, Professor Rasias, and Dr. Angelopoulos. Please, we can see, I think we can watch the video.
Hello, my name is Maria Hadisimeon and I'm going to present you our work entitled Trifluoralin Polluted Soil Treatment Using Nanosecond Pulsed DBD Plasma. The main objective of this work is to examine cold atmospheric plasma, CAP, as an advanced oxidation process for the efficient remediation of soil contaminated with herbicide trifluoralin. For this purpose, we tested a novel DBD reactor, the one that you can see in the picture. We investigated the role of parameters such as pulse voltage, electrode gap, and soil type on the trifluoralin degradation efficiency. Soil pollution is a serious environmental issue as it affects not only the environment but also human health. The main sources of soil pollution come from industrial and agricultural activities or improper waste disposal. In this work, we examine trifluralin as an organic pollutant in soil. Trifluralin is herbicide which has high soil persistence resulting from its low mobility, and according to WHO, trifluralin is neurotoxic, embryotoxic, carcinogenic and highly toxic to aquatic organisms. So we examined dielectric barrier discharge (DBD) plasma for soil remediation from trifluralin. DBD is the electrical discharge which is produced between two electrodes separated by a dielectric barrier. The schematic diagram and the picture below show a typical construction of a DBD. During CAP treatment, there are several reactions taking place, such as the dissociation of nitrogen, oxygen, and water molecules present in the discharge zone. Therefore, many reactive species are produced, which contribute to the degradation of organic molecules in soil. Here we can see the schematic representation of the experimental setup. It consists of three main parts. The first part is the DBD reactor, which is going to be described in detail. The second part is a nanosecond pulse high voltage generator. The pulse voltage and the current are monitored using a digital oscilloscope. And the third part is a gas supplied system where dried compressed air is injected into the reactor with a constant flow. We developed a novel cylinder-to-cylindrical grid DBT reactor. The main idea in this reactor layout is the potential of in situ soil remediation as the plasma discharges are produced inside the pores of the soil. In the picture above, we can see a cross-section of the reactor where the polluted soil is packed between the electrodes. The reactor consists of a cylindrical stainless steel high voltage electrode surrounded by a cylindrical quartz tube serving as the dielectric barrier. The grounded electrode is a stainless steel cylindrical grid attached to the cylindrical quartz. So discharges are produced inside the pores, in contrary with other CAP systems where they are produced above the soil surface. The plasma experiments were conducted with some constant values such as the flow rate of the injected gas and initial concentration of the pollutant, whereas we made a parametrical analysis of treatment time, pulse voltage, soil mass and soil type. After the experiments, trifluralin was extracted from soil using methanol and analyzed with HPLC. Here we can see the electrical characteristics of the nanosecond pulsed DBD. Typical current and voltage waveforms are presented in the figure at the left. Each waveform has a sequence of nanopulses where the rise time of the main pulse was very short at 4 nanoseconds. Also, the peak pulse current was 100 ampere, and therefore there was an extremely high instantaneous power of 1.8 megawatt. In contrast with the high instantaneous power, the mean power dissipated in the system was very low. During parametric analysis of the system, we investigated the effect of pulse voltage on trifluoralin's degradation efficiency. Both plasma treatment time and pulse voltage enhanced trifluoralin's removal from soil. Also, degradation efficiency increased rapidly at short treatment time and gradually stabilized at its examined voltage. It is noteworthy that degradation efficiency surpassed 95% after 10 minutes of treatment, when pulse voltage was 22 and 27 kilovolt. We also made an exhaust gas analysis which indicated the existence of nitrogen dioxide reactive species. We observed that the increase of pulse voltage affected positively the production of this species in the gas phase, as we can see in the diagram on the right. Three different high voltage cylindrical electrodes with different diameters were tested, as it is shown in the picture. These diameters correspond to different electrode gaps. Electrode gap is the distance between the two electrodes, and in this reactor layout it is equal to soil thickness. For the removal of trifluoralin, it is observed that degradation efficiency is increased with the decrease of soil thickness. Therefore, degradation efficiency of trifluoralin reaches almost 100% after 10 minutes of treatment when soil mass is 6.1 grams, whereas at 16.7 grams the corresponding degradation efficiency is almost 80%. Degradation efficiency of the herbicide was examined in two different soil types. The first soil type was a clean sandy soil, and the second one was a loam filled soil. We observed a decrease in degradation efficiency when loam soil was used in comparison with the sandy soil. For example, after 2 minutes of treatment, the degradation efficiency in sandy soil was almost 80% and declined to 40% in loam soil. However, at 10 minutes, a respectable degradation efficiency of 74% is achieved in loam soil. The organic matter present in loam soil and its lower porosity and smaller grain size may affect negatively the penetration and diffusion of active species in soil. The role of active species was also investigated with the use of scavengers, which indicated indirectly the role of the reactive species in the process. Potassium dihydrogen phosphate was used for the inhibition of electrons, whereas sodium azide was used for the inhibition of singlet oxygen. The presence of both scavengers inhibited the degradation of trifluralin in soil, confirming that both electrons and singlet oxygen play an important role in the removal of the herbicide from soil. To conclude, high pulse voltage and the increase of nitrogen dioxide in the exhaust gases resulted in the increase of degradation efficiency. Trifluralin's removal is feasible even at higher soil thickness, whereas electrons and singlet oxygen play an important role on its removal from soil. And finally, the combination of plasma discharges inside the pores of the soil with nanosecond pulses resulted in a highly efficient system for soil remediation. Thank you very much for your attention.
Thank you very much, Maria, for a nice presentation.
Thank you for your attention.
Are there any questions at this point? Okay. Nobody has a question? Maria, I would like to ask you a question. In your mind, is the approach you are, I think you have demonstrated, the potential of your approach, but in your mind, is your approach financially sustainable?
We have tried this in a lab environment. So the reactor is quite small yet, but the plan is to put more quantity to scale up. It is not. I think it will not be very expensive in comparison with other technologies because it has high energy efficiency.
So you bank.
But the thing is to scale it up.
Yes, yes. Okay, thank you very, very much, Maria. That was very, very interesting presentation and work you guys are doing. And our regards to all of your colleagues. And I hope, you continue attending the session. At this point, I would like to introduce the next speaker, Mr. George Kolkos. Am I pronouncing it right, Yorgo, George?
Yes, it's nice.
Okay.
In English.
His co-author is Professor Anastasia Steriadou, and the title of the presentation is Implementation of Hydroseeding Methods as Bioengineering Techniques for Soil Stabilization and Reclamation Projects, I think you want to say, right? Or projections?
Projects is right.
Yeah, it's misspelled, here.
Okay.
Thank you. Please go ahead and try to keep the time, okay? 13 minutes.
Yes, of course. Thank you very much. I hope you see my share screen. Okay, good afternoon to everyone. My name is Georgios Kolkos. I'm a PhD candidate at the Faculty of Forestry and Natural Environmental Sciences of the Aristotle University of Thessaloniki. And on behalf of our Institute of Forest Engineering and Topography, I would like to thank the organized committee of the conference for this opportunity to present our research. The title of my presentation is Implementation of Hydroseeding Methods as a Bioengineering Techniques for Soil Stabilizations and Reclamation Projects. During the construction of roads and in general other technical works, serious soil disturbance occurs. On the left picture, you can see steep slopes which were created during the construction of a forest road. And on the right picture, you can see forest opening up after the completion of the installation of a natural gas pipeline. The main issue, which is caused by soil disturbance, is erosion. Soil stabilization and restoration of the disturbance is achieved by various methods, which can be classified as purely technical, such as the retaining walls and the metal textiles, and also there are many planting works. One very well-known planting technical method for soil stabilization is hydroseeding. Hydroseeding is a mixture of water, seeds, fertilizer, and mulch materials, which are usually created from wood residues. And this mixture is sprayed with the appropriate mechanical equipment on the disturbed surface in order to accomplish the restoration of the vegetation and so the protection from the erosion. The main benefits of this method are the support of supports the regeneration of the vegetation, and also it is considered a very environmental friendly and also a low-cost method in contrast with other technical methods. Here you can see a small video where during the implementation of a hydroseeding, the mixture that I was described before is sprayed along the slope. Sorry. And here there are an area before and after the implementation of the hydroseeding. The kind of the proportions of the materials which constitute the mixture varies and depends actually on the type of the soil disturbance, especially if the soil is earthy or rocky, the vegetation zone, the altitude of the area, the climatic conditions, the exposure of the slope, the existing flora, and of course the organic matter and the nutrients in the soil. The aim of the present research was to demonstrate hydroseeding as a bioengineering technique for soil stabilizations. 3 different cases were analyzed. The analysis took into account all the factors that contribute in the success of the hydroseeding, and the surface coverage was calculated as an indicator in order to compare these 3 methods. The first case was from soil disturbance that occurred from pipeline construction. The second was for hydroseeding for road constructions, actually the slopes that were created during the road constructions. And the third, the second method was from road constructions, and before the implementation of the hydroseeding, there were previously put inside the slope an organic geotextile. And the third method was also from road construction slopes, where there were placed before the implementation of the hydroseeding synthetic geotextiles. This table shows all the factors that contribute in the hydroseeding. Here we can see the location. The 3 cases differ based on the vegetation zone, differ in the type of the soil, the aspect, the incline of the slopes. If there was previously a geotextile was there, the amount of the seeds which were placed inside the mixture, the amount of the organic mulch, the fertilizer, if there was any soil stabilizer, and of course, if after the first implementation of the hydroseeding, any maintenance works took place. Here on this table, you can see the grass species that were used in each case. The selection of the grass species was made based on the factors of the previous table, and especially the vegetation zone and the altitude of the area. All these kind of species are well known for using in grazing. It is necessary, if we want to implement hydroseeding, to be that we can supply a large amount of seeds. So the most of these species are are kind of grass that is used in grazing. Here, sorry. Here we can see the results of the case one after 1 year. The implementation of the hydro seeding there took place on the regional district of Florina. And after 1 year, we had this result as it is shown in the right picture. Here, we see the results after 3 months since the implementation of the hydro seeding in case 3 and in cases 2. The left picture is from case 2 and the right picture is from case 3. So after 3 months, we have this kind of the result. The diagram here shows the percentage of coverage with grass of the area through time. As we can see, in case 1, the coverage of the area with grass managed to reach 100% after 12 months. On contrast, on case 2, there was 90%, and on case 3 was 60%. It has to be highlighted that all the hydro seedings took place on spring. So after 6 months, during where we have the autumn rainfalls, the cover of the area was overcame the percentage of the 30%. Sorry, of the 60% after 6 months. Thus, the erosion issues have been prevailed with the implementation of this method. In general, we have managed to, we can say, sorry, that the soil preparation is extremely necessary in order to have a successful hydro seeding. During the organizing of the construction work, it has to be, we have to secure the topsoil, which after the completion of the configuration of the slopes, we have to put it around the slope in order the grasses have the necessary soil and the organic matter for their vegetation. Repeated hydro seedings would also be a lot of beneficials. If there was any repeated hydro seedings after, we can say, 1 month or 2 in the previous case that we were analyzed, the percentage of the grass coverage will be much better. The maintenance of the hydro seeding is actually a very important factor. Usually, there are no irrigation works or fertilizing works in the slopes and the hydro seedlings in the slopes where we implement hydro seedlings. Thus, we extremely suggest that any studies must have special irrigation and fertilized works for so that we can have a more successful result. The use of mulch materials was less in cases 2 and 3. This is an important factor because when geotextiles are used, it is necessary to use less mulch materials in order to help the sunlight reach the seeds and provide the necessary temperature for their growth. So at this point, I would like to thank you all for your attention. And if you have any questions, please ask. Thank you for your attention.
Iraklis?
Yes. Thanks a lot, George, for this interesting presentation. Any questions? No, actually, I have one, George.
Yes,
can you hear me?
Yes, please.
Yes, I just want to know your opinion about the use of this method for restoration or reclamation of mines, and if this you think is an efficient one?
Absolutely, it is very efficient in mine reclamations. I know from my work that in two places in Greece, in the gold mines in North Chalkidiki and the mines in the island of Milos, this kind of method is used very much. It's really helpful where the soil is disturbed and it has problems with with much heavy metal concentrations, and where the organic matter is really less. So we extremely have many cases shown that we use hydroseeding in mine reclamation.
Thanks a lot, George.
I jump in for another question, George. Right on that point that you made, that was the question I initially wanted to ask anyway. If you guys have ever monitored the vegetation post-mortem, after the fact, after you have planted and you have succeeded, to see what's the uptake. I'm sorry.
Sorry, I didn't hear the.
Yeah, if you have monitored and analyzed the material, basically the vegetation, to see for any uptake from the soil, any uptake of heavy metals or maybe other contaminants, and the part B of the question is, since you have placed this in road embankments, could it be also, have you checked or do you know if the vegetation would absorb, let's say zorb, because I don't know if it's adsorb or absorb, it could be both, I guess, any of the contaminants coming from the roadway?
Okay, thank you very much for your question. In general, hydroseeding, it's a slope stabilization technique. It is a way that we can ensure that the seeds that we plant along the soil were certainly that they will grow. We have actually it's a mechanic technique. With the grass should be grow in less than 10 days. In 10 days we have the first signs of vegetation, and in 3 months the most percent of area of the soil must be full with grass. So we want to protect the erosion. Your question is very, very nice because it really matters what kind of species we use. I said in my presentation that in general, we use grass species from grazing. This is a factor because we can find them in the market and we can supply this large amount of seeds that we need. The kind of the species that we use, actually, it's from the physiology of the plants. Each species can absorb specific kind of heavy metals. I have seen some other research, especially in Canada, where they use hydroseeding in order to have a grass that they absorb specific metals from the soil. So it's actually used in reclamation and more remediation for remediation scopes.
Yeah.
But it's really hard to find in the market this kind of seeds in the supply that we need it. So for that reason, it's not actually takes place in Greece.
Okay, but this could be, further research, meaning that you understand that I'm very familiar, with your approach, and I understand the mechanical part very well, and I understand that you basically the first task is to control erosion. However, the way things are proceeding, you understand that the other questions will come up, will surface up no matter what. both the question of what the uptake phytoremediation is from the soil, from the soil cover, if there is such an issue, as Dr. Panagiotakis mentioned, as well as on what you can uptake and not have it go through the soil from the contamination coming, let's say, from roadways. Thank you very, very much, George.
Thank you very much.
And Iraklis, please.
Παρακαλώ.
Yes, let's move on to our next presentation by Ms. Galani, Ms. Anastasopoulou, Professor Nitsopoulos, and Professor Mamais. The title of the presentation is In situ bioremediation of groundwater polluted with hexavalent chromium under anaerobic condition. I think that Adriani is here.
Thank you, Mr. Panagiotakis.
The floor is yours.
Good afternoon. My name is Andriani Galani, and I'm a PhD candidate at the National Technical University of Athens in the School of Civil Engineering. Before I proceed with the presentation, I would like to thank, excuse me, I haven't shared my screen yet.
Yes.
Yes.
Thank you.
Yes.
That's okay.
That's okay.
Okay, yes. Okay. Before I proceed with the presentation, I would like to thank Ms. Petra Anastasopoulou, Professor Konstantinos Nitsopoulos, and Professor Daniel Mamais for their contribution to this research paper. The subject of this paper is in situ bioremediation of groundwater polluted with hexavalent chromium under anaerobic conditions. Groundwater and soil contamination by chromium has increased dramatically with the advent of rapid industrialization and urbanization. Chromium frequently appears in many industrial waste from activities such as electroplating, metallurgy, tannery and textile, paints and pigments, and glass making. In the environment, chromium is usually encountered in two stable oxidation states. Trivalent chromium is more stable, forms relatively insoluble substances, has low toxicity, and it is not able to cross cell membrane. On the contrary, hexavalent chromium is very soluble, highly toxic and carcinogenic, causing a great threat to human health. One solution to detoxify aquifers is the reduction of hexavalent chromium to trivalent. There are many techniques towards that purpose that could be categorized into two major groups. Based on the location of the remediation, we have in situ and ex situ methods, and based on the treatment method, physicochemical and biological methods. Biological reduction of hexavalent chromium or bioremediation is a cost-effective, environmentally friendly, and sustainable method of remediating polluted groundwater in situ, which can be achieved by stimulating the indigenous microbial populations in soils and groundwater through the addition of various carbon sources. Another advantage of bioremediation is the enhancement of the availability of iron in groundwater. However, the success of bioremediation is affected by soil characteristics, temperature, and the microbial community. The purpose of this research was the study of bacterial anaerobic respiration and electron transfer relevant to hexavalent chromium removal. It aims to experimentally investigate the potential microbial hexavalent chromium reduction to trivalent chromium by biostimulation through the addition of two carbon sources and assess the potential of hexavalent chromium reduction by a coupled biotic-abiotic pathway with the addition of an iron source under anaerobic conditions. In order to simulate closely natural conditions, we conducted microcosm experiments with soil samples, groundwater, and the microbial community that had been acclimatized in the presence of hexavalent chromium under anaerobic conditions. Individual microcosms were constructed in 160 milliliter serum bottles with the equivalent of 20 grams dry weight soil, to which 80 milliliters of liquid amendments were added. The soil and the liquid amendments originated from an anaerobic parent culture. 5 microcosm treatments were set up and duplicate bottles were prepared for each treatment as shown in figure 1. The bottles were flashed with a mixture of nitrogen and carbon dioxide and sealed with gray butyl rubber stoppers and aluminum creams. After preparation, each bottle was spiked with a selected electron donor. Ferrous sulfate heptahydrate was chosen as the iron source. As organic electron donors, molasses and emulsified vegetable oil, or EVO, were selected because of their different properties. Molasses is a readily biodegradable waste byproduct of sugarcane refining, whereas EVO is a slowly biodegradable soluble substrate that ferments to acetate and hydrogen. One amendment did not receive any electron donor, and 2 were spiked with a combination of a carbon and the iron source, as shown on this slide. The parent culture comprised of a slurry of subsurface material in groundwater and 30 milligram tough SS per liter of activated sludge from an anaerobic reactor. It was developed and maintained in the laboratory and was exposed to low hexavalent chromium levels and fed weekly with a reduced anaerobic mineral medium, a selenium tungsten solution, yeast extract, a vitamin solution, and 30 milligrams per liter butyrate, which served as the electron donor. The solid retention time was 45 days, and the acclimatization lasted for 135 days. Each bottle was applied once with an electron donor at the beginning of the experiment. The experiment then proceeded by spiking the bottles with hexavalent chromium every 2 to 3 days without the addition of any other reducing agent. The initial chromium concentration was 2 milligrams per liter per bottle in all batch tests. The day of the spike, liquid samples were withdrawn at predetermined time intervals and analyzed for hexavalent chromium. To describe the transformation kinetics of hexavalent chromium, pseudo first order kinetics were used. Time profiles of every duplicate of the batch experiments were produced on linear plots of ln C over C initial versus time, with the slope of the linear trend line being the K constant. In figure 2, you can see an example of this. You can see an example of the time profiles used. These diagrams are from the molasses experiment on day 2. All rates reported on the next slides are mean values of the duplicates of all the batch tests performed at the selected time intervals, accompanied by standard deviation. At first, we studied the influence of the electron donor, and as it can be observed from the biotic control experiment, hexavalent chromium reduction is possible even without the addition of a reducing agent through endogenous decay. However, compared to the biological enhancement experiment with the addition of molasses and EVO, the removal rates are 2 to 6 times slower, which indicate that microbial growth enhancement was achieved. In the molasses experiment, the reduction rates show a faster decreasing pattern as was expected. Molasses are depleted within 20 days and thereafter chromium reduction proceeds due to endogenous decay at a lower removal rate. In contrast, the results from the EVO amendment indicate that although during the first 30 days of the experiment, The differences between the rate constants of molasses and EVO are negligible. During the next time period, the removal rate increases by 78% and then decreases rapidly. This finding is on par with the expected effect of EVO on the microbes as a slowly depleted electron donor. On this slide, we can see the influence of the addition of ferrous iron. In figures 4 and 5, the kinetic rate constants after the addition of ferrous iron are presented. Addition of ferrous iron can double the removal rates during the first 10 days of the experiment. However, for the rest of the experiment, between the 10th and the 16th day, the kinetic rates of the bottles amended with carbon source and divalent iron were similar to those containing only the corresponding carbon source. In conclusion, the results verified that the addition of an organic electron donor can increase the rate of hexavalent chromium reduction up to 6 times in comparison to the natural soil capacity rates. The 2 carbon sources employed for biostimulation exhibited similar hexavalent chromium reduction rates during the first 30 days of the experiments. However, the slow biodegradable organic substrate exhibited higher longevity. Furthermore, the addition of a carbon source and a ferrous iron source can increase the removal rate of hexavalent chromium significantly during the first days of its addition. However, it does not have a long-lasting effect on hexavalent chromium reduction. Concluding this presentation, I would like to mention that this research is co-financed by Greece and the European Union through a doctorate research program and implemented by the State Scholarships Foundation. Thank you for your attention.
Thank you very much, Adriani. That was super interesting, as you know, especially for us, the co-chair's presentation, as we have been doing a lot of work on hexavalent chromium. I would like to ask you a question. Mm-hmm. And the question is, okay, there is an issue of the longevity of treatment, obviously, here. Mm-hmm. You mentioned it also in the conclusions. Is there something out of the work that you guys did and maybe have not presented today, or out of other work of other people? Is there any alternative there to have a long-lasting, in other words, you know, to follow up a remediation scheme, mm-hmm. but to have a long-lasting, longer, let's say, lasting than what you've researched, namely EVO and the ferrous iron source?
Okay, from the work of other people and from the Superfund sites from the US EPA, I have found that the molasses, yes, is depleted very quickly, and actually, if we try to implement the method on the field, we have to inject a molasses solution every few months, every 3 to 4 months. For the EVO, we have to inject the sites every 2 to 3 years. So EVO does have a longer lasting effect. Now for the iron, the problem with the iron is that it doesn't have, it's not very mobile in groundwater. It's not very mobile in the soil. It depends on the soil characteristics more appropriately. And we used the soil that had the pH of 8.3, 8.4. So it didn't have not almost immediately. I mean, most of it was used for the reduction of hexavalent chromium, but a small percentage was definitely rendered inactive because of the pH of the soil and the water.
Due to oxidation?
It was oxidized, yes. Yes.
Thank you very, very much, Adriani. At this point, is there any other question, first of all? Okay, at this point, I would like to introduce the next speaker, Mr. Markus Hort. And he's going to also be talking about the very dear to both of the co-chairs, subject, PFAS removal, performance of novel technologies for treatment of firefighting water and groundwater. You can introduce your co-authors should you choose to. Markus?
Yes.
The floor is yours.
Yeah, thank you. Thanks a lot. So today I would like to present to you the result of a project that we conducted looking into novel treatment technologies for the treatment of PFAS in firefighting wastewater and in impacted groundwater. I would like to make a big thank you for the co-authors as well, as you see on the screen. If I can. Yes, so this is the outline of my presentation. First, I will just introduce CONCAWE, which is the association which I'm working for. And then I will move into a short introduction, present to you the materials and methods applied, go into the results and discussion, and finally end up with the conclusions and an outlook. So CONCAWE, for those who don't know, is the science division of the European Petroleum Refiners Association. It was established in the 60s and now includes most of the oil companies operating in Europe. The CONCAWE mission is threefold, and it's about improving scientific understanding and also assist in developing policies and legislation at the EU level, as well as informing decision-making for our membership. And we perform science in many areas, for example, in fuel technology and air quality. But today I'm working for the water, soil and waste group within CONCAWE. So this is in the area where I will make my presentation. So as a background, most of you know about PFAS, so I will not dwell too much into it, but it's a very complex group of substances, and there is many different types of PFAS, and the number keeps increasing more or less daily. And why it's relevant for the refining industry is because it has been applied in firefighting foams since about 1970. And the background and the driver for this study was to look into PFAS in terms of groundwater remediation and firefighting wastewater treatment, as firefighting in refineries can result in large volumes of firefighting wastewater, which can impact both surface waters and groundwater when the treatment and containment of the PFAS is not performed appropriately. And conventional treatment systems have shown that they're not very efficient in removing PFAS. And while there are technologies applied to remove PFAS, there are also recently alternative treatment options available. So the driver for this study was to look into these alternative, more novel treatment options that are available, and look into how effective they are in treating PFAS in both impacted groundwater and in firefighting wastewater. So looking at the materials and methods, we took two samples. One was from an impacted groundwater from a site in Europe, and the other one was a firefighting wastewater obtained from a firefighting training site. The groundwater sample was also spiked with benzene in some selected experiments to study the potential effect of petrochemical co-contamination. And we selected the technologies to be studied based on a previous CONCAWE report that was issued in 2020, and that looked on several factors in terms of the treatment technology. For example, the water composition of the water to be treated, and the reported treatment efficiency for a wide array of PFAS substances. Within this particular study that I will present today, it was done in 2 parts. I will focus on the first part, which is the experimental phase, where we studied the separation of PFAS from the waters in a laboratory setting. There was also a more comprehensive technology evaluation done, but I will not have time to discuss that today, but that's part of a Concur report. As an experimental overview, we studied different types of treatment technologies, both sorption technologies and physical separation technologies. And we used the granule activated carbon, or the GAC, as the benchmark technology. And then to be able to, that has been widely studied. So we wanted to have that as a benchmark to compare with other more novel technologies. And we studied the different technologies both in batch mode and continuous mode, as you can see in the table on the screen. So moving on to the results and discussion part. First of all, the composition in both of the samples, not surprisingly, varied greatly, where the groundwater contained a lot of different types of PFAS, and the firefighting wastewater was more dominated by a precursor substance. Also, the concentration of the 2 samples were very different, which was not unsuspected, since the firefighting wastewater is coming directly after the application of the firefighting foam, and the impacted groundwater is something that is coming, was obtained outside the refinery site. So moving on to some of the highlights from the testing that was performed. In the batch tests, applying both sorption technologies and flocculation, it was observed that the granule activated carbon and one of the sorbents, so a clay-based sorbent, showed the highest removal of PFAS, as can be seen on the right-hand side. Sorbent dosages were below 100 milligram per liter, or even 10 milligrams per liter. And in those dosage levels that appeared to be sufficient to obtain a relevant treatment performance. Data that is not shown here, but is available in the report, is showing that the longer the carbofluorinated chain, the more effective all the sorbents became in removing that the PFAS. Also, in the groundwater experiments, which was spiked with benzene, overall the clay-based sorbents performed similarly to the unspiked experiments, whereas the polymeric-based sorbents, their performance was increased, meaning that more PFAS was actually removed, and the flocculation performed poorer when it was spiked with benzene. From the groundwater batch tests, we also attempted to determine the sorption coefficients for specific types of PFAS species. And also here you can see a list showing which technology exhibited the highest sorption coefficient for different PFAS species. And they were determined by fitting an isotherm at a given concentration at 1 microgram per liter. So you see on the right-hand side the example of PFOA in the groundwater sample or during the groundwater treatment, and here you see that the granule activated carbon exhibited the highest sorption coefficient. Still continuing on the batch tests, but this time with the firefighting wastewater, it was observed that the flocculant was the most effective technology in reducing the sum of PFAS. It was also the most effective one at lower concentration. However, all sorbents require dosages in the range of grams per liter, and that was also true for the flocculant in order to be effective. So it seemed from this data that both potentially flocculant can be suitable for direct firefighting wastewater treatment, but none of the sorbents seem to be very suitable for doing direct firefighting wastewater treatment. Moving on to not to the sorbents and not to the flocculants, but to another novel technology, the foam fractionation with ozone and air. In these tests, due to a limitation of the project, full process optimization was not feasible due to excessive foam formation. However, some observations could be made, and those include that the ozone treatment appeared to be more effective than the air treatment. And moreover, that the foam fractionation led to the concentration of PFAS in a small volume and a significant reduction of PFAS in the treated water, as can be seen on the table on the bottom right-hand side. Overall, it was difficult to conclude definitely due to the limitation in the operating conditions. However, it seems to be showing potential at least as a pre-treatment technology. And finally, of the novel technologies assessed was the nanofiltration, which was performed only on the firefighting wastewater. And from these tests, it was shown that the nanofiltration technology was able to remove PFAS efficiently during the first hours of operation, but when operating for longer period, as can be seen in the red circle on the right-hand side, the passage of PFAS through the membrane increased. So moving on to the conclusions and outlook of the study. First, I will focus on the conclusions on the performance testing and dividing them in each per technology. So on the sorbents, it was concluded that all the tested sorbents were capable of removing at least 85% of the sum of PFAS from the impacted groundwater, and that was independently if they were spiked with benzene or not. A direct firefighting wastewater treatment is not advisable, since it requires both a high loading of the sorbent applied, and it doesn't give a complete PFAS removal. Out of the sorbents, the granule activated carbon appeared to be the most robust one, motivated by both the results from the batch tests, which were the data that I showed today, and also based on results from the continuous mode tests, which the data I did not show today due to the interest of time. Regarding flocculation, it appeared to be able to efficiently pre-treat the firefighting wastewater. Probably a polishing step will be needed after the flocculation step in order to achieve a sufficient removal of the PFAS. Regards to the flocculation and the groundwater, it showed an incomplete removal. On the foam fractionation part, it showed that you could separate the PFAS by concentrating it in a foam matrix, but overall it was considered insufficient as a standalone treatment for the tested water, but showed potential as a pre-treatment step. Regarding nanofiltration, we observed that there was very good initial removal, exceeding 95%, but fouling occurred after about 10 hours of operation. So that led us to conclude that firefighting wastewater treatment with nanofiltration might require pre-treatment in order to prolong the service life of the membrane applied. And the overall conclusions of this study were that physical separation technologies appeared to be suitable to at least pre-treat heavily contaminated matrices. So, for example, firefighting wastewater, while sorption techniques were more suitable to treat cleaner matrices. So, for example, impacted groundwater, which contains lower concentrations of both PFAS and also by other co-contaminants. A general solution that can be applied to refinery wastewater, firefighting wastewater treatment, or for groundwater remediation was not it's not available based on the results of this study, and really a case-by-case assessment must be made. But it's likely that a multi-barrier approach, so first a pre-treatment step followed by a polishing step, is appropriate for most sites, and also depending on the matrices that is studied or is needed to be treated. As an outlook and next steps for this work would be to study even shorter chain PFASs, since which with a carbon number lower than 4, since that was not analyzed within this study, as it was not part of the analytical suite of that was applied. Also, it would be nice to study a wider spectrum of PFASs. So in this project, we studied 20 different substances, but as you know, there is plenty more PFASs out there. So as a next step, it would be interesting also to study more substances. Finally, on the outlook part, we did not study reactive treatment technologies such as incineration, and they also have been, according to the literature, showing promising result. So that would also be an interesting technology type to study. I only today had the time to, due to the interest of time, to focus on some parts of the work that was done in this very comprehensive piece of work. But there is a Concur report available on our website, and that contains much more of the details of the work that was conducted within this project. And it also contains lookup tables for all the assessed technologies, and that's done in order to guide practitioners in order to select the most appropriate technology for their specific site and their specific matrix. It also includes all of the performance results and discusses consideration of practical application of each technology when it's applied in the field. I would like to say a big thank you to all the people that they were involved in making this project a success. So it involved people from KWR in the Netherlands, the Expertise Centrum for PFAS in the Netherlands as well, the University of Amsterdam, and the Concur Soil and Groundwater Special Task Force. So thank you a lot for your attention, and I'm happy to take any questions.
Please. Irakli.
Yes. So thanks a lot, Marcus, for this very interesting presentation on this very, very hot scientific issue. There is a great discussion currently ongoing on PFAS remediation. Is there any question? Yes, please, Professor Dermatas.
I have some questions. First of all, the first question is, I understand the use of petrochemical co-contamination. Why did you pick benzene?
I mean, there is a lot of different co-contaminants that you could pick. We picked benzene because we thought that was the most well-representing compound, and that we couldn't study them all. So due to the limitation of this project, we had to pick one really, and we thought that benzene was one that you can find in contaminated groundwater or in the firefighting wastewater. So we thought that was something representative to study that study the influence of co-contamination. It's not a complete approach, but it was something that we found that was representing that to some extent at least.
No, thank you very much. I understand it's not a complete approach, and it's impossible almost, I think, to have a complete approach, because first of all, there are so many PFAS, as you said, and then it really is significant, the co-contamination. I mean, I know that there's a lot of work on that, and it's just, fragmented. There's no consensus, if you may. That's why I asked you the question. If you guys reached the consensus of how you picked benzene, you answered, then how did you pre-select your sorbents?
Yeah, so we had a review done, which I referred to in the materials and methods, and based on that, we also looked on what was practically available to do in a lab setting, and the ones that we thought, based on the conclusions of that report, which seemed to be the most promising for this application.
So you applied some kind of criteria for the sorbents, because as you said in the presentation, the materials and methods, you said you went a literature review, but I was asking about what type of criteria, if you have them, or what was the most significant criterion to choose?
Yeah. So I think the most significant criteria were that where it had been shown that they had been successfully applied in other cases. Yeah, and then also if they were if you could scale them down to a lab setting in a sufficient way, because in some of the cases I know that you cannot. It's difficult to grind the material down to be able to perform lab experiments of them. So that was also a limitation.
I'm just picking up on your answer here. So grinding is not going to be applied on the full scale, because surface area plays a tremendous role. So I didn't understand the last part you said about the grinding. So
no, but
you wouldn't grind. I mean.
No, but in some cases for the granule activated carbon, sometimes they would not fit in the lab setting, so they had to be slightly modified in order to fit that.
Yeah, changes the effectiveness also. The other thing I wanted to ask is, in your view, because you compared, safe concentrations, you mentioned sufficient removal, you mentioned 95% removal, more than 80%. What's sufficient removal in terms of, because I would imagine it would vary according to the specific compound. And
yeah,
they haven't been studied all. I mean, is it now the best we can do, kind of, best available practice or the basis, or is there a basis that I'm not aware of?
I mean, there are no, in at least in the regulatory context, there is not. There might be some local limits to comply to, but there is not a really consensus, I think, on what is the sufficient removal. So we kind of based it on if it was achieving a 95% removal or 90% removal, and on that basis we said that the removal was sufficient to qualify the technology as being either suitable for a specific case or not suitable for a specific case. So depending on the firefighting wastewater or the impacted groundwater.
But you haven't been able to match, because we have the same difficulty as we try to study this animal that's called PFAS, this complex animal. There's no guidelines that you came across or that you used targeting specific concentrations, let's say.
I mean, yeah.
I'm sorry.
No.
No. Okay. And the last question should be very simple. It's just I didn't grasp it during the presentation. You had the batch experiments. The continuous experiments were also absorption experiments, or
Yes.
they were column or something else?
Yeah. Well, they were done in column testing. So, but still in quite a small scale.
Okay.
But in a continuous mode.
Thank you. That's all. Thank you very, very much. It was very interesting, your presentation, Mark, and good luck with any future work. And I hope, Marcus, and I hope, we can maybe, collaborate on that too, since we're also having an active group here doing research on that.
Thanks a lot.
Thank you.
Okay, let's move now to the flash presentations. The first one is by Ms. Ayomi Posi on groundwater flow dynamics in shallow coastal plain sands aquifer in Abyssinian area, eastern Dahomey Basin, southwestern Nigeria. I think that we can watch the video.
Hi everyone, my name is Anne Joseph. Today I'll be discussing on groundwater flow dynamics in shallow coastal plain sands, southwestern Nigeria. Groundwater recharge and discharge are critical to global hydrological cycle, but are not noticeable. Groundwater flow in the subsurface is driven by changes in energy. Water flows from high energy areas to low energy areas. And the type of geological materials that are present and the length of time water is in contact with those materials are the two fundamental controlling factors on water chemistry in drainage basins. The rate at which water migrates between groundwater and surface water provides a significant pathway for chemical transfer through different processes like precipitation, acid-base reactions, sorption, ion exchange, and so forth. Hence, it is necessary for us to understand the groundwater flow to aid proper development, management, and exploration of groundwater resources, so that one will be able to understand the contamination levels and how to analyze how water gets contaminated through human activities. The scope of my work revolved around Abyssinian community, which is one of the highest population in southwestern part of Nigeria. The inhabitants rely on groundwater extracted from hand-dug wells. You can see southwestern part here, Abyssinian community here, and it's more widened here in the red spot. The overall objective of this my research work was to determine the hydrologic head distribution, analyze the groundwater flow, delineate the research and discharge and recharge areas, and identify wells around these zones to determine the pattern of distribution of ions around this zone. So from our results, we analyzed dissolved anionic species such as chloride, carbonate, sulfate, and nitrate to delineate this sort of areas. So you can see from this map, the map below underneath that which overlies the whole or other components is the map of Abeson. And these flow directions are representing the flow of water. Flow out means recharge and flow in means discharge. So we can see that we had a couple of recharge and discharge places. And we found out that chloride was higher than the carbonate concentration in these recharge areas, and conversely, which happens for the discharge zones. Hence, this provided a means for us to identify these recharge and discharge areas, which were amongst these pointed purple areas where the well and groundwater samples were taken. I'm also able to determine the contamination levels of this to potential salinity, which we discovered that there was no, the water was not really saline, but was unsuitable for irrigation through different analytical methods. Thank you for listening.
Okay. At this point, I would like, before we move to the next flash, I just noticed there's a question on the Q&A. It didn't show up, probably my mistake, I don't know, but it didn't show up before, and it's for Adriani. Adriani, are you still here?
Yes, I'm still here.
Yeah, the question is, will the reduction also work on higher chromium-6 concentrations? Have you determined an upper chromium-6 concentration limit that the chromium concentrations work? Is by Florian Lab.
So we knew from previous work in the laboratory that above the level of 2 milligrams per liter, a higher concentration of hexavalent chromium are toxic for the microbes. So yes, it would not work.
So there is an upper limit of 2 milligrams.
It is an upper limit. 2.5. So yes, we didn't try anything above that.
Thank you very much, Adriani. Answered that question, and we go to the next presentation by Professor Muhammad Al-Swayan, modeling the evaporation flux from shallow sandy aquifers. Is also a flash presentation. Please go ahead.
Good morning, ladies and gentlemen. The title of my presentation is Modeling the Evaporation Flux from Shallow Sandy Aquifers. I am a professor in the Civil Engineering Department at KFUPM. This is a university in Dhahran, Saudi Arabia. In this paper, a mathematical model based on Richard equation and using the soil hydraulic properties was developed in order to understand the influence of the soil depth to the water table on the maximum potential evaporative flux, in which water flows through the unsaturated zone. The model was applied on shallow aquifers consisting of different types of sandy soils with different hydraulic properties that were collected from the literature. Simple equations were developed in order to estimate the maximum evaporation from shallow sandy water table aquifers. This is in a nutshell what we have here. The conceptual model is shown here, where we have a profile or a section view where we have the water table and some soil. Here we call it sand, but it can be any type of soil. At a depth, the depth below from the ground surface to the water table is variable, and we have evaporation. And the mathematical model that will be used here is Richard equation, which appears on the right here, where HC here is the capillary pressure, and Q is the evaporation rate. This is the steady evaporation rate. KW is the unsaturated hydraulic conductivity of the wetting fluid, which is water. This equation is solved using different types of soils and determining the maximum Q, which will occur obviously when we assume that the capillary pressure here is very high. These are the one diagram that shows the result of different soils. Of course, it is clear that the depth has significant influence on the evaporation. The higher depth, the lower the evaporation rate. But this is quantified and equations were developed. The details are in the poster and in the paper. Thank you and have a good day.
Okay, let's move to the next class presentation. This is biodegradation of anthracene by an Antarctic fungal strain, a member of genus Alternaria, by Dr. Gerginova, Dr. Stoyanova, Ms. Peneva, and Professor Alexieva. Let's watch the video, please.
I cannot hear anything. Okay, this video unfortunately doesn't have sound. It was sent like this, so we just have to watch. Everybody, unfortunately. Okay, let's move to the next video, I would say, because it's kind of, next video is by the same group. I hope it's exactly the same group. It's a study of the potential Rhodococcus strains for degradation of the pesticide glyphosate Roundup, which is a quite a famous, and toxic pesticide. Please go ahead. It's the same group. I just hope that this time we have the sound on, that they have recorded sound. Okay, same problem, huh? I don't hear anything. Same as before. Okay, and it's hard to see anyway.
Dimitri, this is posters anyway, so the audience can also
check them out. Okay, so at this point, Dr. Panagiotakis and myself, we would like to extend one last request for any questions, and I will check also the general audience questions. I don't see anything yet. Okay, since we don't have any more questions, I would like to thank everybody for their participation. I would like to specifically thank, obviously, the presenters and all co-authors for having put up their work, for us to share and ponder. It was a very interesting session with many interesting presentations. And I would like you to just keep on going on the next, session on the virtual room. Iraklis, do you have to say anything?
I just want to say again, thank you to Margot de Kleen for accepting our invitation, and to also to invite our speakers to join us as well to our tomorrow's workshop on contaminated soil management in Greece, learning from EU and the international experience. Thank you all.
Thank you all.
Bye-bye.
Bye-bye, Margot. See you tomorrow. Okay?
Bye-bye.
Bye-bye. Thank you very much again. Thank you. Indeed. So let's leave. Ευχαριστούμε. Ευχαριστούμε.
Okay, we are ready, I assume, to start, right? Okay. Okay, excellent. So good afternoon to everybody. Welcome to section 18. My name is Konstantinos Plakas. I am associate researcher in the Center for Research and Technology, Hellas, which is one of the major and leading research centers in Greece. And I will chair this session entitled Sustainable Water Management in the Mediterranean Technological Solutions Demonstration and Deployment. This is a special session organized by a cluster of 5 European Neighborhood Instrument Cross-Border Cooperation Mediterranean Program funded projects related to water and sanitation, namely Aquacycle, MEDIS, Manawara, Nagoment, and PROSIM, with the aim to present innovative technological solutions and approaches towards increasing water efficiency and encouraging the use of non-conventional water supplies in the arid and the semi-arid areas of the Mediterranean basin. Before starting this session, I would like to thank, on behalf of all projects representatives, Mr. Mofer Hint from the Palestinian Wastewater Engineers Group, Dr. Alberto Carletti from the Desertification Research Center and Department of Agricultural Sciences of the University of Sassari in Italy, Dr. Fabio Massi on behalf of Erythra Company in Italy, Mrs. Valeria Loni on behalf of the Institute for University Cooperation Onlus in Italy. Would like to thank the organizer of this major CEST conference, especially Dr. Eleftheria Klonja and Professor Dimitris Lekas, for accepting and embracing our proposal to organize this event, for their excellent collaboration and assistance. Our session includes 8 presentations, 2 on behalf of Aquacycle project, 2 of Menawara project, 1 from MEDIS project, and 1 from Nagoment project, as well as 2 presentations from Spain and Greece, presenting results from novel national R&D projects. I would like first to ask Mrs. Irene Morel Rodriguez, Senior Expert on Environment and Climate Change of the Joint Technical Secretariat of the NSCBCMED program, to open this session by introducing the program and its strategic goals to the audience. Mrs. Rodriguez, the virtual floor is yours. Is Irene connected? Okay. Irene, I don't know if you can hear us. Maybe you are muted. If not, we can try later and continue with the program.
Yes.
Okay, then we start with the first presentation.
Okay.
By Ms. Inmaculada Paulo Lopez, a senior researcher in CEMAT Plataforma Solar de Armería in Spain, as well as a member of the Spanish Aquacycle team, presenting the work entitled Application of Two Different Strategies for the Simultaneous Disinfection and Decontamination of Urban Wastewater using Solar Photophantom and Solar Hydrogen Peroxide Processes. Please, Inma, the floor is yours.
Thank you, Kostas. Thank you for the nice introduction. I'm going to share now my screen. Let me know if you can see my screen now. Yes? Okay. Okay, thank you. So today I'm going to present a summary of a study, an experimental study, titled Application of Two Different Strategies for the Simultaneous Disinfection and Decontamination of Urban Wastewater using two types of different solar processes, namely photophantom and hydrogen peroxide in combination with natural sunlight. The contents of this presentation is a short introduction, then the objective, then the main material and method that we have used for obtain the results that we will see later, and finally some concluding remarks. So the context of this studies focus on the application of innovative processes for enhancement the secondary effluent of urban wastewater treatment plant from considering two different targets. The inactivation of microbials, according also to the new European regulation on wastewater reclamation and reuse, but also on the elimination of contaminants of emerging concern, which although they are not yet contemplating in this regulation, they are containing in the secondary effluents of wastewater treatment plants and must be eliminated before to reuse in any type of final point or activity to avoid any risks related with these contaminants. So regarding the processes that we have investigated, the first one is, as I told you in the title, is the solar photophantom. This process is an advanced oxidation processes which are defined like any chemical treatment procedure designed to remove efficiently organic compounds and microorganisms from water by using oxidation reaction and that permit the generation of hydroxyl radicals. These hydroxyl radicals are reactive oxygen species, non-selective, that can react with almost all organic compounds in the water, leading eventually the total mineralization. So in particular, solar photophantom can be driven by natural sunlight, and it consists on the mix of iron salts, like you can see here in the main two reactions of these processes, iron salts, hydrogen peroxide, and the natural sunlight. When we have these three compounds in the water, it generated a catalytic cycle where hydroxyl radicals are generated. This process is highly efficient in the degradation of contaminants and also in the inactivation of microorganisms, but it has the important drawbacks to be implemented at real scale. And this is the efficiency of these processes is maximum at acidic condition at a pH lower than 3. But of course, for a real implementation, the cost of modification of the water pH before and after the processes is prohibitive to be implemented, as well as the sludge production. So some possible solution that has been investigated during last years in this sense has been the use of chelating agents to keep the iron in solution the time enough to generate hydroxyl radicals to promote the degradation of contaminants and the inactivation of microorganisms. One of the most efficient chelating agents is the ethylenediamine and disulfinic acid, or the EDDS, the short name. These processes using the EDDS has been reported and testing like a highly efficient on the removal of organic contaminants. Other process that we have investigated in this study, related also with the reagent using in the solar photophantom, is the hydrogen peroxide. This process is not a real advanced oxidation processes because it's not generate hydroxyl radicals in combination with natural sunlight, because the wavelengths that reaching the Earth's surface are not strong enough to seize the hydrogen peroxide molecules. Therefore, this process has never been used for the degradation of contaminants itself, but also some years ago, we observed, some researchers started to observe that this process was highly efficient for the inactivation of microorganisms. And this is based on internal cell damage generated by the free diffusion of hydrogen peroxide, the mechanisms that have been explaining to explain the inactivation of microorganisms, and we will see later more in detail. So the objective of these processes was to investigate the efficiency of both processes individually at neutral pH, operated using a very low cost system, namely raceway pond reactor, to simultaneously remove a mixture of contaminants of emerging concern, some pharmaceuticals including, and also different type of bacteria, Enterococcus fecalis, Enterococcus fecalis, and Salmonella enteritidis. And 3 different set of experiments were carried out in using simulated urban wastewater spiked with this mix of contaminants, such as like you see here now. The hydrogen peroxide sunlight alone, using a 50 milligrams per liter of this oxidant, the solar photophantom with EDDS, using a molar ratio of 1:1 and 0.1 millimolar of iron EDDS, and also including the same concentration of hydrogen peroxide. And then we combine both treatment in a sequential treatment to try to improve the capability of this process. Sorry, this concentration, I should say that it was, they were previously optimization in previous work. So the main materials and methods used in this study was the simulated urban wastewater, which is a model of the secondary effluent of a wastewater treatment plant, mainly characterized by around 50 milligrams per liter of dissolved organic carbon. We use different analytical devices to determine determine the concentration of the contaminants of emerging concern using UBL system. Also some spectrophotometric methods to determine the concentration of iron and hydrogen peroxide along the treatment time and the process. And the solar radiation was monitored also during all time using an UV pyranometer. Regarding the bacteria, they were spiked at a concentration of 10 to the 3 CFU per milliliter and they were monitoring along the treatment time using the standard plate count method, using selective agar medium for that. The initial concentration of each CEC, it was 100 milligrams per liter. So this mix was added to the reactor. Here you can see a picture of the reactor, which is located at all the experiments were done at the Plataforma Solar de Almería, which is a research center where I belong to. It's located at the southeast of Spain. For those who don't know this center, and here you can see a picture of the solar devices that we use for performing all the solar treatment. And among all these devices, we use also this very low system, the raised wave pump reactor. As you can see, it's a very simple system, and it adapts from the use of growing microalgae. But some years ago, we started to investigate the capability of solar treatment using this system, because it is a very low cost treatment system. In particular, the raceway that we use for this study, it is characterized by a working volume of 90 liters with 50 centimeters of liquid depth. Along all the time, the water is homogeneous by using a paddle well that permit to keep this homogeneous mix during all the treatment. So let's go with the main results. First of all, I would like to show you the results obtained with the treatment where we use only hydrogen peroxide in combination with natural sunlight. On the graph on the left, we see the profiles of bacteria inactivation. We have in the y-axis the concentration of bacteria, the time, the treatment time, in the x-axis. In the top, the solar UVA accumulated along the treatment time. And in the right, the hydrogen peroxide concentration detected and monitored along the process. It is important to focus on the treatment time of each bacteria, and we observe that specifically, the Escherichia coli reached the detection limit in less than 20 minutes, and the more resistant one is the Enterococcus fecalis. This is a common result because Enterococcus fecalis is a gram-positive bacteria that should be more resistant to most of the oxidative treatment. If then we focus on the graph in the right, related with the profiles of each contaminant of immediate concern, we observe that most of them still remain at the same of the initial concentration, except diclofenac that show degradation along the treatment time, but associated mainly to the photolysis, the effect of this compound. So we can conclude from these results that the process is feasible for the inactivation of bacteria, but is not efficiently for the degradation of CEC. The explanation regarding the CEC, the no degradation of CEC, is because no hydroxyl radicals are generated during the process. But here is a summary of the mechanisms of inactivation of bacteria. This is not experimentally yet determined, but the scientific community accept that the inactivation mechanisms of bacteria is based on the free diffusion of hydrogen peroxide from the external medium to the internal cell through the cytoplasmatic membrane. When inside of the cell, it reacts with hydrogen, with the iron that is naturally occurring inside the cells. Then this reaction generates hydroxyl radicals by similar reaction of the typical phytoplankton, generating hydroxyl radicals that affect all the internal compounds of the microorganisms, accelerating a lot the inactivation of this bacteria. So then here I show you the results using the solar phytoplankton with EDDS and also remember, with the same concentration of hydrogen peroxide. If we focus on the graph related with the bacterial inactivation, we observe that the treatment time required to obtain the inactivation of Escherichia coli is longer than in the case of hydrogen peroxide alone. And even in the case of the more resistant microorganisms, the Enterococcus fecalis, the bacteria remain constant. So no inactivation of this bacteria we observe along treatment time. In the case of the CEC degradation, we observe quite opposite the results, because in a very short treatment time, we obtain more than the 80% the degradation of the sum of the all CEC. So clearly, that means that the process of solar photophotosynthesis with EDDS is not a good option for the inactivation of bacteria, but is very efficient on the degradation of CEC. The reason to obtain this lower efficiency regarding to the inactivation of bacteria is explained due to the hydroxyl radicals generated are faster during the first minute, but this is not enough to obtain a good inactivation. So other actions are acting during the rest of the treatment time. And this is firstly the light scattering that can be generated by the water brown color and the higher turbidity due to the iron precipitation that happen along the processes. This iron precipitated may act like a screen, making the bacteria protected from the action of solar light, so reducing the efficiency of the solar effect on the inactivation of the microorganisms, and also the presence of higher concentration of organic matter when we have the EDDS, because it's an organic compound, compared with the concentration of this organic matter in sunlight hydrogen peroxide alone. And it can impact on the inactivation rate of bacteria due to also the competition of the organic matter with the hydrogen, with the hydroxyl radicals generated. So therefore, we decide to combine both processes in a same treatment in terms of applying firstly the sunlight hydrogen peroxide during a certain time or the time needed to reach the complete inactivation of bacteria, and then we add the iron EDDS. So in the graphs we observe firstly the same results regarding the bacteria due to the effect of hydrogen peroxide alone. We obtain the inactivation of E. coli in a very short treatment time, and even the inactivation of Enterococcus fecalis in 60 minutes with no regrowth of bacteria after the processes, which is quite important. And if we focus then on the graph related with the CEC degradation, we observe again the same effect. Almost no reduction of the CEC during the hydrogen peroxide treatment time, but a faster degradation when we apply the iron chelate, obtaining finally a complete removal of bacteria and CEC in around 70 minutes of solar treatment time. So finally, the main concluding remarks from this work is that it's important to evaluate when we apply a process. It's important to be sure that we can obtain the best results, not applying the treatment by only one type of target, because in wastewater we have many different types of targets, and because these results, if we analyze individual targets, we can obtain inaccurate conclusion related with the efficiency of a process. Then the solar photophotosynthesis and the sunlight hydrogen peroxide as standalone processes were not effective enough for the simultaneous removal of contaminants of emerging concern of bacteria in the reactors we use. But the sequential treatment, combining both processes in one single process, in the raceway pond reactor, it was a suitable solution for the simultaneous purification of the water. And also, it's important that before to determine if this process is very suitable and affordable for real implementation, it's important to investigate the same processes in actual secondary effluents, and as well the analysis of other additional parameters such as the bacterial regrowth after the processes, the toxicity created by the applied chemical reagents, and also the treatment cost. And finally, thank you for your attention. And if you have any question, just please feel free to ask any doubts.
Thank you very much, Emma, for the excellent presentation and the very interesting work carried out the premise of Plataforma Solar de Almería. We are very happy and proud to have in AquaCycle project PSA with us on board. I would like to stress that the solar raceway pond reactor technology will be also tested at pilot scale in the 3 demo plants to be installed in Lebanon, Spain, and Tunisia, so as to evaluate in a real operating environment the capacity of this technology to disinfect and together remove the different organic micropollutants that escape the previous constructive wetlands and anaerobic digestive processes, so as to have a clear water for reuse. Inma, I don't see any question in the virtual chat.
Okay,
I will
just
wait
say that
1 minute if there is any question.
Yeah, I would like to say that this approach that we started to investigate some months ago, we are going to apply also in the AquaCycle project as part of our very successful collaboration. And for sure, I hope the results will be nice. And also, it's important that finally, I would like to highlight the importance of not only apply a single processes, if not to investigate all the boundary parameters that will finally define if the process is suitable and affordable for real implementation.
Exactly. I would like to ask the presenters if there is any question for Inma. Alberto, Fabio, Rula, or someone else, or else we can move to the next presentation. It was clear from Inma that the quality characteristics play a crucial role, the turbidity, the organic content, right, Inma?
Yes.
Which we, of course, investigated in the context also of AquaCycle project, how detrimental they are to the effectiveness of this technology.
Yeah.
Yes.
So thank you very much, Inma. We can move to the next presentation by Dr. Alberto Carletti, Senior Researcher in the Certification Research Center and Department of Agricultural Sciences from the University of
There is a question from
sorry, Alberto, yes.
From Mr. Not from me, in the chat, there is a question.
In the chat, so there is a delay. I don't see in the chat, to tell you the truth, any question.
It is Mr. Hamadi.
Yes, for me, there is two worries to use this to disinfect wastewater with the photo fountain. Is one how to adjust the pH in the inlet, and the second is about the regrowth. That we know that even with UV light, we have regrowth of bacteria in the storage of water, and when we will use it for irrigation, we have to deal with the regrowth of bacteria.
Yeah, regarding the pH, in fact, the use of the iron chelated is specifically to avoid the need to adjust the pH. So it's when we use the solar photophantom with EDDS, we apply at the natural pH. We don't modify the pH. The typical or the conventional solar photophantom, the most effective way is at pH 3. But of course, this is not affordable in a real scale. For this reason, we move to the use of the iron chelate that permit to work at a neutral pH. And regarding the bacterial regrowth, this is a quite important point. So when you investigate and look for the inactivation of total bacteria, you must be sure that this occurs. If the process is not good enough and you remain some bacteria in the water, it could regrow. So for this reason, it's important to know that all the microorganisms and especially the fecal contamination are killed and cannot regrow after that. And with the treatment time that we apply and with the processes that we apply, we know that after 24 hours and even 1 week after the application of the treatment, there is not bacteria regrowth itself. Other things is that you have another recontamination. And for this reason, also the storage is important in this kind of processes. But if you don't have a recontamination of the water, the bacteria, after to be inactivated, will not regrow. But it's important that you reach that point.
Amadi, I think the answers provided by Inma are quite adequate. It's understood that we don't modify the pH. It's a natural pH applied by applying selective iron chelate agents for the scope. If there isn't any other question, I propose to move to the next presentation, if you all agree.
Okay. Thank you.
So, thank you one more time, Inma. So, Dr. Alberto Carletti, the floor is yours. Just to remind that Alberto is also a coordinator of Menawara project, organizing this special event. And his presentation is entitled Fostering the Non-Conventional Water Use in Agriculture in Mediterranean Countries, the Menawara Project.
Thank you, Costa. Good afternoon to everybody. I'm going to share my screen. Okay. Okay, can you see my presentation? Okay. Okay, yes. I'm Alberto Carletti. I'm a researcher at the Desertification Research Center at the Department of Agricultural Science at the Sassari University in Italy. As I'm coordinator of the Menawara project. And today I will present you an overview of the project and mainly focus on data related to the non-conventional, the treatment of non-conventional water for its reuse in agriculture. The Mediterranean region represents an hotspot for climate change, and it is considered as one of the world's most water-stressed areas, where some countries have less than 1,000 cubic meters per capita per year. These countries are witnessing of increased pressure on fresh water, expressing a growing gap between supply and demand of water and deterioration of the surface and groundwater quality. The agricultural sector accounts for more than 70% of total water demand, and to which water losses during conveyance and distribution are added. In the perspective of a more efficient and integrated management, the use of non-conventional water as drainage water and treated wastewater represents a valid alternative to reduce imbalances between resource availability and demand, also contributing to the promotion and implementation of the circular economy concept, not only from the point of view of water availability, but also from that one of nutrient and energy recovery. However, the agricultural sector has to adapt to the new political and institutional framework, both at national and international level, which is taking place and is characterized by a synergy between agricultural policies, environmental policies, and conservation of natural resources. The Menawara project is one of the 5 demonstrative projects funded by EU under the NECBC MED program within the topic aimed to support innovative and technological solutions to increase water efficiency and encourage the use of non-conventional water supply. The project started in September 2019 and will last 36 months. The joint challenges consist of providing additional resources, in particular non-conventional water, by recycling drainage and wastewater to reduce the pressure on conventional water, and by rationalizing water use practices to increase water use efficiency in agriculture. The total budget is around 2.9 million euros, out of which 2.6 as a EU contribution, corresponding to the 19% of the total budget. The overall objective is to contribute to increase the water availability for agricultural purposes through the use of non-conventional water to reduce the pressure on fresh water. And we have 3 specific objectives: improve water use efficiency of non-conventional water for irrigation, improve the quality of non-conventional water to use in agriculture, and strengthen the non-conventional water governance by disseminating and capitalizing innovative and technological solutions implemented by the project. The consortium includes 6 partners from 5 Mediterranean countries: Italy, Spain, Tunisia, Palestine, and Jordan. The lead beneficiary is the Desertification Research Center of the Sassari University. CIAM is an international organization which since many years has worked in all Mediterranean areas. NARC is the National Agriculture Research Center of the Ministry of Agriculture in Jordan. WeWork is an Italian NGO which participates in Menawara with its branch in Palestine. ONAS is the governmental agency which manages all the wastewater treatment plants in Tunisia, and CENTA is a public foundation under the Andalusian government, which has a strong expertise in wastewater treatment technologies. The project are proposing different solution, sustainable, clean, environmental friendly solution based on the use of non-conventional water, in particular treated wastewater and drainage water, and innovative techniques. In Menawara, it is fundamental the application of the fit to purpose approach, which is a new important concept in water use, which is production of reclaimed water quality that meets the needs of the intended end users, always in accordance with the quality standard and regulation established in each intervention country. Another important pillar of the project is the involvement of stakeholders at different levels since the beginning of the activities. The proposed solution can be classified into 4 categories: minor intervention in some existing wastewater treatment plants to improve their efficiency, low-cost pre- and post-treatment system realized in these plants, especially as a tertiary treatment, sustainable and effective irrigation system adopted to improve water use efficiency and crop yields by using treated wastewater. This system could be innovative mini sprinklers with high resistance to clogging, or subsurface and surface drippers adapted to treated wastewater. Finally, managed aquifer recharge system realized by using the forest infiltration area technique to mitigate the nitrogen water concentration in the aquifers of the Italian intervention area. A participatory and knowledge sharing approach is being adopted with relevant stakeholders, including national authorities, water agency, and associated partners. My colleague Rula Kadra will give more details on this in her presentation later, but in few words, I can say that demonstration sites will act as a living lab and learner spaces where public-private stakeholders will identify constraints and opportunities for a productive use of treated wastewater in irrigated agriculture, design, equip, and put in operation the demo sites, and evaluate the innovative irrigation trains' impacts on efficiency, productivity, and on groundwater. The intervention will be implemented in 4 wastewater treatment plants in Tunisia, Shutrana 2 and Burst Wheel near Tunis, Korba and Kelibia in the Cap Bon Peninsula. In Palestine, the plant is in Bein Dajan near Nablus. In Jordan is in Ramta in the Irbil region in northern Jordan. At the experimental center of Senta near Seville. In Italy, the MAR system will be implemented in the nitrate vulnerable zone of Arborea in central western Sardinia. The technological intervention have been designed according to these steps. First, identification of the quality standard and regulation established for the reuse of non-conventional water in agriculture in each project country. Field surveys to assess the efficiency of the identified wastewater treatment plants and to characterize the non-conventional water quality according to the national quality standards. The preliminary design for minor intervention, pre and post treatment in the selected plants, and the design of field system in Arborea. Field and market surveys for the definition of the executive design and tender procedure for the implementation of the work. At the moment, the works are under implementation in Jordan, while tender procedures are ongoing or will be launched very soon in Spain, Italy, and Palestine. In this following slide, I will show you the main characteristics of the technological intervention. For example, in the plant of Shutrana 2 in Tunisia, the treatment line consists in train from the existing secondary clarifiers and control basin, based on a filtration processes using pressure sand filters and a disinfection stage through ultraviolet radiation. In Jordan, the treatment line reclaim the secondary wastewater, treated wastewater coming from the plant of Ramta and in an existing storage pole. The tertiary treatment train is composed of a filtration treatment through the pressure sand filters, followed by a disinfection stage by ultraviolet radiation and maintenance chlorination. Compared to the other plants in the project, the current technical status of the plant in Bein Dajan, Palestine, doesn't allow to the production of treated wastewater, which is in line with the national quality standard and the farmers' needs. For this reason, the different solutions have been designed to improve the efficiency and the performance of these plants. This intervention includes minor intervention, innovative pretreatment compact system for screening, sand removal, and degreasing of wastewater. All the function will be controlled by a control panel. The post-treatment is based on a filtration process using pressure sand filters and disinfection by hypochlorite. At the experimental center of Senta in Carrion de los Céspedes in near Seville, the design treatment train is based on different kinds of constructed wetlands. They are followed by a storage pond with ultrasound treatment and pressurized sand filtration. In the nitrate vulnerable zone of Arborea, the managed aquifer recharge system based on the technique of forest infiltration area will be tested as a best practice to contribute to reduce the nitrate groundwater contamination for the sandy phreatic aquifer in the Arborea plain. This system will be implemented in on a surface of 0.4 hectares and supplied by drainage water pumped by from an existing dewatering pumping station. The design consists of 6 parallel recharge trenches placed between rows of poplar tree and equipped with a passive treatment system, which consists of a mixture of inert materials, gravel and sand, and organic materials, eucalyptus wood chips, which function is to promote denitrification processes, attenuate organic and inorganic contamination, and prevent clogging processes at the infiltrating surfaces. In this image, you can see a simulation of the FIA system in Arborea. So in conclusion, we can say that design and technological intervention will significantly contribute to improve the agricultural sector, taking another step in its process of adaptation to the new political climate scenario and to the improvement of well-being of local farmers. FIA systems be integrated in the Sardinian Basin Management Plan as possible tool to mitigate the groundwater degradation, not only in the Arborea Plain, but also in similar hydrogeological contexts in Sardinia, but in the future, it can be replicated in other Mediterranean countries. In the long term, the engagement of associated partners and other national authorities due to their institutional function will ensure the replicability of the action, generating multiplicative effects. Thank you. This is our content of the project. I would like to point out the address of the web platform of the Menawara project, where we are uploading many information regarding best practices on non-conventional water treatment around the world. We are uploading also the results and of the activities of the project, the living labs, and so on. Also a collection of legislation in different countries and so on. I would like to invite you to join this platform. Thank you for your attention.
Thank you very much, Alberto, for the very nice presentation, presenting Menawara project and the different best practices proposed in the context of your project. I see some similarities with AquaCycle that will be really interesting to brainstorm on the results and check and verify the optimum solution, especially for the sake of the farmers, especially in the arid and semi-arid areas of our Mediterranean basin. Is there any question for Alberto? Actually, I'm checking two chats, one in the Zoom and one in the platform of the conference. That's why there is a delay, between the two platforms. Let me check if there is any question for you, Alberto.
I would like to say only that our cluster in the NECBC model will give the chance for us to join results, to explore future collaboration also in our project countries. Is a first step for a future initiative and the progress in these countries.
And to tell you the truth, the actual results on the pilot test to be carried out by the different projects will be available not later, but the beginning of 2022 or somewhere between spring of 2022 due to the emerging condition posed by the COVID-19 pandemic. There were many delays with the tendering and procedures for implementing the different demo setups. I don't know if something similar is also for you in your project.
We are all in the same situation. We accumulated a big delay due to the COVID situation in all the countries. We are finalizing all the procedure to realize the pilot site. Yes.
Is there any other question for Alberto? So, Alberto, I don't see any question for you. So I propose that we continue with the next presentation. Will be carried out by myself, representing the AquaCycle partnership. So let me share my screen. Mm-hmm. Can you all see my screen? I think. Okay. Okay. So in this work co-authored by the partnership of AquaCycle project, we present the distinct features of an eco-innovative system, wastewater treatment process scheme, and its characteristics for the cost-effective and with maximum environmental benefits treatment of urban wastewater in small and medium-sized communities in the Mediterranean region. At the beginning, I will briefly refer to the challenges in the Mediterranean region, particularly in the MENA countries, in regards to the availability of water as the most essential natural resources, and how AquaCycle project addresses these challenges. One of the novelties is the development and operational evaluation in real environment of the process scheme with the acronym APOC, for the cost-effective treatment of urban wastewater for safe reuse with minimum operating cost and maximum environmental benefits. Next, a brief description will follow of the 3 APOC demonstration units to be installed in Tunisia, Lebanon, and Spain to test and validate the efficiency and cost effectiveness of the hybrid system. And finally, we'll summarize the key performance indicator to be assessed during the operating demonstration of the 3 demo systems. So it is well known that the Mediterranean area is one of the most vulnerable to water crisis and to the impacts of global warming. Despite some encouraging progress, current water use efficiencies in the drinking water and irrigation sector still represent a challenge for the area. This has been recognized by the ENI program, which has set among the utmost, I would say, priorities for the NSBC-Med cooperation area, the improvement of the water efficiency, with a particular focus on agriculture, as well as increasing the water supply through non-conventional water resources. It is true that 40% of the cities in the Mediterranean area with a population between 2,000 to 10,000 inhabitants are not connected to any wastewater treatment plant, while a substantial number of wastewater treatment plants installed in the Med area have proven unsuccessful copies of treatment system operating in the Western world, while these systems are often unsuited to address the local challenges of wastewater treatment, especially at small scale. Therefore, treatment of municipal wastewater is commonly exploited throughout the region. Also, sorry to minimize this, the Mediterranean is also affected by a generalized economic slowdown, which is driven, among others, by the consequence of the COVID-19 pandemic, the reduction in investment, and a lack of sufficient employment opportunities. So the critical water stress situation is also visualized in the graphs of this slide. According to a recent work carried out by the University of Montpellier and Plan Blue, based on global climate projections and water use scenarios inspired by national reports, by 2050 horizon, the water stress could increase over the whole Mediterranean basin, notably because of a 30 to 50% decline in freshwater resources as a result of climate change. In addition, under a business as usual water use scenario, total water withdrawals are projected to double on the southern and eastern rims. These worrying trends indicate the need to develop mitigation scenarios. In this context, AquaCycle project funded by the EU under the NSBC-Med program aspires to change the paradigm of viewing wastewater as an unsafe effluent to that of an abundant all year round resource that has multiple uses. AquaCycle is a 3-year project that has started on 1st September 2019 and is expected to end by the end of August 2022, although we expect an extension of our project of more or less 6 months. due to the delays posed by the COVID-19 pandemic. Aquacycle Consortium includes in total 7 partners from Greece, Spain, Malta, and Tunisia and Lebanon. 4 are research centers. 1 is a university, 1 is an SME, and 1 is a major entity for the water sanitation in Spain. I'm referring to Zamur. There are also 4 associate partners that compose the advisory board of the project, coming from Greece, France, Algeria, and Morocco. This challenging and together and exciting project was, I would say, expected to finish by September 2022, but we expect a projection of 4 months. And has a total budget close to 2.9 million euros. So, Aquacycle in a nutshell. In the context of Aquacycle, we aim to promote the operational application of an eco-innovative wastewater treatment system, which is based on natural processes and renewable solar energy, and test this process scheme by installing 3 pilot demo units in Tunisia, Lebanon, and Spain. Through our project, we aim to promote the water circular economy by providing an alternative supplementary water resource that is readily available all year round. At least 10 showcase and knowledge transfer measures will be implemented for the training and skills development of local actors, including decision makers, professionals, and young scientists. We aim to improve also water governance through the active involvement of the local communities in the development of wastewater treatment and reuse action plans. 3 PPGIS-based municipal action plans are expected, fully owned by the local communities, and with a set target for the reuse of up to 900,000 cubic meters of treated effluent. Finally, we aim to prepare the ground for investment in the new APOC technology, increase the employment opportunities, and stimulate the long-term endogenous economic growth. So investment plans based on detailed techno-economic documentation with potential funding instruments and investors will be pursued in the context of our project. The acronym of APOC systems stands from anaerobic digestion, photocatalytic oxidation, and constructive wetland, which are the 3 components of the eco-innovative wastewater treatment system proposed by the Aquacycle project. The anaerobic treatment and the constructed wetland are 2 mature and commercialized technologies with wide applications in the wastewater treatment market that are combined with a novel solar disinfection photocatalytic oxidation process presented later previously by Dr. Immaculata Lopez Polo towards the treatment of municipal wastewater at a level that satisfies the most stringent standards for reuse. In this slide, you can see a generic APOC plant layout. The component of this layout, of course, are strictly indicative and can be modified by the plant designer depending on the effluent characteristic and the treatment objectives. Specifically, different types of anaerobic digestion reactors can be implemented as well as constructed wetlands, whereas the post-treatment that composed by the novel solar raceway pond reactor is a distinctive feature of the APOC system. The main APOC features is that the system can treat wastewater for both standard discharge and for reuse application. The system has the ability to treat domestic wastewater from secondary to tertiary level and can be generally a community managed system. Also can be constructed according to effluent wastewater characteristics as already mentioned. It provides an effective solution for ecologically sensitive areas. Can be implemented using local skills and know-how to provide context-specific sanitation services and get optimum efficiency of the system. Can provide a renewable also energy source by the produced biogas from the anaerobic digestion process. Depending on the demand, technical modifications can be made and biogas can be generated. based on the organic content to supply energy. It can provide also a solid bioproduct, the anaerobic digestate that can be used also for land fertilization. And finally, the APOC effluent can be used for urban uses, industrial uses, and other agricultural uses and groundwater discharge. The distinctive feature of the APOC technology, which is of course to produce clean water that is reusable and safe for the environment, make it also eco-friendly, efficient, and cost-effective, as it is based, as you have seen, on natural system. It uses less chemicals. It runs on renewable energy. I'm referring to solar radiation. It produces biogas, fertilizer, and a clean water for use in agriculture, in domestic, industrial, and other applications, while the constructed wetlands thrive as a habitat, as also an ecological tourist attraction, aside from being a climate change mitigation measure. As already mentioned, 3 APOC demonstration units will be installed in Tunisia, in Spain, and in Lebanon, with a treatment capacity that varies from 5 to 20 cubic meters per day. In the case of the demo plant in Spain, this will be implemented in the premises of the Blanca wastewater treatment plant operated by Zamur. The feed flow rate will be 5 cubic meters per day. The anaerobic digestion will consist of an upflow anaerobic sludge blanket reactor, while 2 constructed wetlands will be connected in series, one subsurface vertical wetland and one subsurface horizontal wetland. As already said, the photocatalytic oxidation part to tertiary treat the effluent of the constructed wetland will be common to all 3 demo units and will consist of a solar photoreactor in the form of a roseway pond reactor. The demo plant in Tunisia will be constructed at the premises of the wastewater treatment plant of Ben Zaidan to be operated by CEST. The feed flow rates will be decided, but probably will be close to 15 cubic meters per day. The anaerobic digestion reactor will be a high rate USB reactor. The constructed wetland in this case will be 2 subsurface vertical flow constructed wetlands operating in this case in parallel, as well as 2 subsurface horizontal flow constructed wetlands working in series. Again, the photocatalytic oxidation part will be the solar raceway pond reactor. The demo plant in Lebanon will be established at the premises, on the area, of the Dekura, will be operated by the Lebanese University. The feed flow rate will be 5 cubic meters per day of a domestic sewage coming from a residential community close to Tripoli. The anaerobic digestion will consist of a 4-chamber anaerobic batch reactor, while in this case, the constructed wetland, we will test the performance of an innovative horizontal flow aerated constructed wetland. The demo plant in Spain is under construction. The demo plant in Tunisia currently is under signing the contract with the winning contractor, while more info about this will be presented tomorrow in the context of a flash presentation of the paper number 650, and advise you also to attend this presentation, while the demo plant in Lebanon is under design procedure and launching the tender soon within the next month, hopefully. In this slide, you can see some photos of the ongoing construction works of the demo plant in Spain. Actually, the APOC demo plant will be, as already installed in the premises of Blanca wastewater treatment plant, operated by Zamor. In this demo case, the effluent of the existing, there is an existing USB reactor will feed with a feed flow rate of 5 cubic meters per day, a system of 2 constructed wetlands that are connected in series. Due to the vertical wetland intermediate flow needs, we have also a buffer tank of 3 cubic meter capacity from which the anaerobic digestate liquid will be pumped to the vertical wetland. And the effluent of the two constructed wetlands will be finally led to the solar photoreactor in the form of the RPR, which consists of open channels as the photos that already shown by Ima in her presentation. Here you can see also photos of the ongoing construction work of the APOC demo unit in Spain, and especially of the constructed wetland and of the pumping station. All APOC system will be demonstrated with real municipal influence in an operational environment with the aim to assess the effectiveness of this particular technology towards determining the optimum operating parameters to be recommended later for full-scale systems. The key performance indicators to be assessed during the operating demonstration are related with the treated effluent quality, with the energy savings and the energy recovery, with the treated effluent reuse potential, and the overall cost effectiveness of this novel process scheme. Also recently, CERT team, with the support of AquaCycle partners, completed and launched an e-training platform, which aims to provide the knowledge in the form of guidance and training of all interested parties, ranging from public authorities and educational organizations, as well as to private entities and professionals involved with wastewater treatment. And I kindly ask you all to visit our platform through the link shown in this slide, and also to express your interest either in the question tab to participate in future e-training seminars to be organized soon by AquaCycle partnership. With this slide, I would like to acknowledge and thank the European Union for the financial support of our project under the NCBCMET program, and you also for your attention. So, thank you very much. I don't know if there is any question from the presenters or the virtual participants of our session. I don't see any question in the chat, either in the conference platform or in the Zoom. Alberto, I will really, as well as Fabio and Monther, would like also to interact with you and kindly invite you to our training seminars to be organized in the context of our project in introducing in more details APOC technology. Maybe in the context of this e-training seminar, we have the opportunity to discuss in more details the technical parts of the three different components of our system. I think Inma already presented some details of the solar as waypoint reactor component. Fab is also expert in national-based solution proposal, and as you have seen, Fabio, many different types of constructed wetlands will be tested in our demo plan units, varying from vertical and horizontal, as well as to other novel aerated constructed wetlands. The scope of the anaerobic digestion is to kind of escape from the typical activated sludge process and having a process that is more easy to implement, especially in the Northern African countries. It's lesser complex and also potential to recover biogas as a mean to recover also energy. I don't know, any question? Sorry, maybe my presentation took longer than I expected. Fabio, you're muted. I don't know if you want to make any question. No. Okay. So if you all agree, we can move to the next presentation by Dr. Rula Katra. She's a senior researcher, international officer in the International Center for Advanced Mediterranean Agronomic Studies, Mediterranean Agronomic Institute of Bari, and member of MENAWARA project. Rura, we are very glad to have you with us, and the virtual floor is yours to present your work. Thank you very much.
Under the project Minawar. Excuse me, I don't know what's happening, but actually I can see a.
Can you try again to share your screen?
Yeah, I will try again, but actually I can see a registered version that I didn't know. I didn't do. Just a second.
Okay, now we can see your screen.
Yeah.
You can press F5.
Yeah, I'm trying. I'm trying. Okay, this is working now. Yeah. Okay. Good evening, everybody. And again, I apologize because as I already said, I'm on a sick leave and I'm not able to put on my camera. Today, I would like just to present a little bit the Living Lab approach that you are exploring under the framework of Minawara project to develop a sustainable wastewater treatment and reuse system in Mediterranean agriculture. As you already know, the Mediterranean is a water poor region, and people have always had to cope with scarcity, developing adaptation strategies to meet their essential needs. However, these strategies or these capacities in place are put to test by a number of challenges that were somehow explained and listed by the former speakers. But I would like just to talk about, for example, the uneven distribution, with 72% of resources are in the north, 23% in the east, and 5% in the south. So the shortfall in quantity also has been compounded by a decrease in quality due to the contamination of surface and groundwater resources in the region. And in the last 50 years, the total water demand has doubled as a result of demographic pressure and from the development of water-intensive activities such as tourism and manufacture. Indeed, most of the water is used in the agricultural sector, which presents high rates of inefficiency. On the top, we can find the impacts generated by climate change. Actually, the shortage of water in the region has been affected by this impact or by through the increasing temperatures and the variations in precipitation. And once again, these impacts have different consequences in the region. Southern and eastern countries are more exposed to desertification, increasing soil aridity, and exhaustion of water sources. Even though the recent severe droughts in the region convey important messages about what could happen to all of us in the future. A third important challenge is related to sustainable water management. Actually, the questions of demand versus supply with regard to water management are very important issues that require special attention in a water scarce zone. And for a long time, the supply management concept has dominated the actions in the region. However, technical solutions alone cannot provide the increasing population with a safe water supply and proper environmental protection. Integrated water resources management that include technical, managerial, institutional, social, and economic aspects shall constitute the first priority among the range of actions to be adopted. A very burning issue in the region is the transboundary water management in the southern and eastern Mediterranean. The transboundary resources that are shared among the countries of the region or with countries outside the region, they constitute the majority of the water resources, including both surface and groundwater bodies. And this is a very burning issue. Major transboundary river basin in the Mediterranean, namely the Jordan, the Nile, the Euphrates, Tigris, are in sub-regions that have experienced severe political tensions. These political circumstances have aggravated past water disputes, which otherwise might have been solved had the political climate been more favorable. So the concerned countries should realize that without cooperation, they cannot address the issues of each country, and the only way out is through the cooperation. And here we have the wastewater treatment in the region as an alternative. Actually, in this very hot environment, the reduction in annual precipitation and accentuated climate change effects, droughts, they have driven the consumers to use alternative resources, such as treated wastewater. It is compatible with the concept of circular economy, as already said, and wastewater treatment and reuse has a significant potential to preserve resources in agricultural irrigation and can also serve as a way to restore groundwater capacities. However, intensifying the wastewater treatment and reuse in the water-poor region requires overcoming a number of constraints related to the quality of treatment, the inadequate legislation, limited social acceptance, and viability issues. Moreover, although the reuse of treated wastewater in agriculture has several benefits that include providing renewable nutrients and organic matter useful for maintaining soil fertility and productivity, it may be associated to several risks if not properly addressed. Therefore, it is worthwhile to evaluate agricultural treated wastewater reuse practices. So, achieving success in crop production largely depends on the adoption of appropriate on-farm management strategies aimed at optimizing crop yields, maintaining soil productivity, and safeguarding the environment. And these challenges are pursuant to a range of themes encompassed by the SDGs, in particular 2.4, which seeks to implement resilient agricultural practices that increase productivity and production and strengthen the capacity for adaptation to climate change, progressively improve land and soil quality, as well as the target 6.3 and 6.4, which seek to improve water quality by reducing pollution, having the proportion of untreated wastewater, substantially increasing recycling and safe reuse, and substantially increasing water use efficiency by ensuring sustainable withdrawals to address water scarcity. I just want to recall over the overall objective and the specific objectives already stated by the objectives of Menawara, already stated by my colleague Alberto. So specifically, Menawara seek to improve water use efficiency, to improve the quality, and to strengthen the governance of the non-conventional water resources, and disseminating and capitalizing innovative and technological solutions. In this perspective, would like to outline the basic concept of living labs and its usability to overcome several constraints that hinder the intensification of wastewater treatment and reuse in agriculture, and to ensure the adoption of innovations is successful, sustained, and scaled. A living lab is critically built on a social process that transforms the knowledge from individual level into an ever wider communities of interaction. It is user-centered, open innovation ecosystem, often operating in a territorial context, integrating concurrent research and innovation processes within a PPP partnership. So within a public-private people partnership. The adoption of innovations is based on processes of knowledge creation and learning, thus enabling the individual to develop personal expertise through guided experience with expert or even with more advanced peers. Living lab approach helps to take into account the needs and address the concerns of different user groups when developing new techniques, technologies, and rules, or adapting the existing ones. And under Menawara, the ENI project, 7 living labs in Italy, Jordan, Palestine, Spain, and Tunisia are under implementation. Menawara living labs are environments meant to provide space for exchanges and to facilitate the creative cooperation between a diverse group of actors seeking solutions to the challenges of a particular locality. So solutions that work, fit to purpose for treating around 2,000 cubic meters a day in each partner country. They are water-oriented and demo-type environment with a cross-sector nexus approach. They involve and engage multi-stakeholders, which include ministries, water utilities, water users associations, farmers cooperatives, research centers, and youth networks. And they avail of a certain continuity, and they provide a field lab actually to create awareness, to share or to build shared understanding. And actually, when applying solutions to new situations, this process requires some form of adaptation to the local context. So this is a matter of and recontextualizing knowledge and exploring which conditions can make the application successful and how the solution can be adapted. And applying new solutions entails a certain amount of risk. So the appropriation process, therefore, must establish some form of validation for a solution. And this is addressed through gathering experiences, informal discussions, collecting scientifically based evidence about the success and impact of the considered techniques or technologies, and getting social support and authorization. And these living labs are implemented in partnership with FENTA, with UNIS, Siambari, NARC, WeWord, GVC, and ONAS, as already stated by Alberto. In what area? They will be implemented in Italy in a nitrate vulnerable zone. The zone is Arborea zone in the center west of Sardinia. In Ramda research station, northwest of Jordan, it's Irbid Governorate. In Beit Dajan wastewater treatment plant in Nablus, and in Spain, in Fenta Center in Andalusia region, and in 2 governorates in Tunisia, in Ariana and Nabil. In these living labs, just to summarize a little bit what has been already said, in Italy, the FIA, a managed aquifer recharge technique, was tested as a best practice to mitigate the groundwater nitrate contamination. And this is our first pilot and living lab. In Jordan, a reclamation train based on sand filtration and UV disinfection of wastewater will allow a reliable irrigation of fodder crop used as animal feed over 5 hectares. In Palestine, an innovative pre-treatment compact system and a post-treatment train will enable to serve 7.5 hectares of fodder, citrus, and olive trees. And in Spain, Fenta will host a treatment train with different types and configurations of constructed wetlands, enabling the safe irrigation of olive trees, while in Tunisia, the treated wastewater plants will be upgraded and will serve 25 hectares of fodder and olive trees, and will allow to recover an agricultural vocated area threatened by water shortage and equality deterioration. In the framework of Menawara, we identified momentum phases for the living lab. Because, you know, Menawara is a project and we had to identify the momentum phases, but this doesn't mean that the living lab has to die after the lifespan of Menawara. So the first crucial momentum is called paving the ground, and it actually debates the water allocation and cropping pattern for treated wastewater reuse, water budgeting, irrigation techniques, legal and governance dimensions, the state of the art of wastewater effluent quality, fit for purpose treatment train, expected effluent quality, and tailored use. And I can say that all of our living labs already implemented the first phase. The second phase or momentum phase is called innovating, inspecting to enabling the change. So once the treatment train is operational and we are in this phase, treatment and reuse train efficiency test is performed. The quality and the reliability of the supply is considered together with the rules for allocation in compliance with equity principles among users and in response to the CROPS requirement and environment safeguarding. This momentum phase shall demonstrate the adequacy of the pilot in terms of actions and results to what was discussed and agreed during the first phase. The last phase is evaluating and validating, and it engaged stakeholders from different pilots in the same country and from different partners countries, just talking about integration and cooperation. And it incorporates results and impacts of at least one irrigation season, and it is intended as a cross-fertilization activity that shall yield lessons learned and best practice, these being the characteristics that differentiate a living lab from a pilot action and effectively allow for a scaling up. Living labs in Menawara and in general, they have potentials and challenges. What are the potentials in Menawara? Actually, involved actors are animated for improving the current situation in the pilot areas by implementing new technology that contemplate hard, soft, and organizational wares in real-world test environment. Awareness on how improved wastewater treatment, reuse, and management generate environmental and economic benefits can be raised at the same time. This is also due to the fact that one core element of living labs is that all stakeholders of a certain environment participate and share their knowledge and experiences. Since the living lab approach focuses strongly on the end users who will be gathered in several meetings and events, present and future responsibilities can be discussed and actions for the resilience of the developed solutions beyond the project duration can be defined. Living labs are supposed to function transparent and non-discriminating, which also enhances the chances of success and improve the communication and innovation processes among stakeholders and enables to support the current women empowerment assessment and inclusion within the agricultural sector and how it can be affected by the new technologies and practices. If carried out properly, the needs and expectations of wastewater users are directly transferred to water utilities and authorities, which in turn have a chance to retrieve feedbacks in a transparent and constructive way. Utilities, furthermore, can benefit from the data to be collected within the living labs and get a more clear and proper view on requested future developments in order to match the needs of the users with the approached overall climate, water, and agricultural goals. Coming to the challenges within living lab, they are usually due to the availability of financial and human resources, as well as organization, management related aspects. This applies for the stability of the living lab itself, as well as the equal participation of every affected stakeholder group. The challenge is to engage all affected stakeholders in the same manner. Otherwise, some stakeholders would take advantage of innovation projects without being part of the solution finding process during the living lab. Future challenges that may weaken the success of a living lab are missing flexibility by the involved stakeholders, the lack of trust, psychological barriers, entrenched structures, missing seek for change among communities and institutional organizations. In Menawara, living labs are new approaches for every participating country. However, some countries already worked with similar instruments such as participation processes. Nevertheless, methods for building a sound basis of trust have to be considered when designing a living lab. When thinking of the open innovation approach of living labs, values such as collaboration, sharing, and co-development are essential. An organizational change within participating organizations and institutions should always be evoked in case these values are not fully represented. And thank you very much for your attention.
Thank you. Thank you very much, Rula, for the excellent presentation and for stretching the usability of the living labs as an approach, as a methodology, especially for those who are not keen in such kind of a methodology, which aim to foster and promote best practices, solutions, and sustainable technologies. To be easily also adaptable to the local communities and stakeholders. I have seen that there were some questions that have been already replied by Alberto Carletti in the chat. Just for the virtual attendees, I would like to transfer the questions made and the replies received. Dr. Fabio Massi from Iridra asks if Menawara expects to observe a decrease of the groundwater nitrates concentration by the MAR process in Italy already during the project monitoring time, and by what amount of water to be infiltrated per day approximately. And the answer was indeed, this is the objective of Menawara project, and according to some infiltration tests already carried out in the site, the project estimates to infiltrate around 500,000 cubic meters per year. Concerning the concentration of the nitrates in the groundwater, considering a recharge period from March to October each year, which I assume this has to do with the cultivation period, right, Alberto? In the southern part of the plain where the pilot site will be implemented, the NRD measured up to 180 milligrams per liter of nitrates in the Santi aquifer. I don't know if Dr. Carletti would like or Dr. Massi would like anything else to comment on this point. Concerning the virtual chat room, I don't see any other question.
I would like only to point out that in Menawara, the FIA system implementation is a demonstrative action, is a small pilot plant on a surface of 0.4 hectares. Then the impact on the polluted aquifer and arborea is very localized. Our objective is to test this technique as a best practice to be replicated also in other parts of the arborea plain with some point with other small FIA system in other point distributed in a homogeneous way in the plain. But we have to carry out this step with this experimental phase to test if especially the passive treated system is effective to reduce to improve the quality of the non-conventional water used for the and to have an impact on polluted groundwater.
Okay, thank you, Alberti, for this clarifications provided.
Yes, Kostas.
Yes, Hamadi, please.
Yes, it's very pertinent to have this question from Fabio on is the system will be only made for decrease of nitrate concentration by dilution, or we will favor the conditions for denitrification or something else?
Okay, yes, the impact on the groundwater quality to reduce the nitrate concentration in groundwater is for dilution, yes. But the other aspect is that the FIA system with the presence of the rows of poplar trees promotes also the nitrification process during the infiltration process. And we combine the passive treated system with the future contribution of the roots of the trees to generate anaerobic condition in the ground and promote the nitrification processes during the recharge, because then drainage water that will be used during the recharge in average as from 40 to 60 milligram per liter of nitrate. Then we have to reduce the concentration of nitrate in the drainage water to recharge the aquifer and to reduce the concentration in groundwater.
Okay.
May I make a question?
Yes, please, Montero. I'm very glad to hear you. You would like to ask something to Alberto?
No, to Dr. Rola, please. If she has a couple of sentences more on the pre-treatment plant, the compacted plant she mentioned in Palestine.
I cannot. Yes, Rula.
Yes, Mother, would you repeat, please?
Yeah, I noticed in your presentation you said something about a compact pre-treatment unit in Palestine, which you are following up in Beit Dajan, perhaps, if I understood you correctly. Could you say a couple of sentences about what technology you are using in this pre-treatment, please?
About the technologies, I will leave the floor to Alberto. I'm more in the treatment, I'm more in the reuse in the agricultural environment and the implementation of the living labs.
Okay.
I can say to Monter only the thing that I show in my presentation related to this compact system to reduce sand removal, decreasing wastewater. I think that if you want to have more details, we can ask to my colleague Isabel Martin that designed with her colleagues in Senta this system, this treatment line where the compact system is integrated. I don't know if Isabel can say something. She should be in the meeting.
Hello. Do you hear me?
Yes.
We can hear you, Isabel.
Okay, hello to everyone, and congratulations for the session and also for the so interesting presentations. Well, related with the pre-treatment to be installed in the Beit Dajan was what the treatment plan in Palestine. The design is a very innovative approach. It's a pre-treatment compact system. And some remark relate with this pre-treatment compact system. I can mention, for example, that the particles to settle is near to 2,000 micrometers, for example, present a modular structure that allows to be assembled directly on the site. The machine can be run manually, or you have the option to run it automatically. It's depending of the operators of the wastewater treatment plant, and also present easy maintenance. more characteristic of this compass system, for example, that principally this, that the assembly directly on site, thanks to the modular structure, the easy maintenance. Also, you can have the option to use the internal view of throw, because pressing scroll bars, bolted, etc. And then, as commented in the presentation, my colleague Alberto, all functions of this pretreatment system can be controlled by a control panel. So this, in our opinion, is a more sophisticated and a more adequate, more modern pretreatment system.
Okay. So sorry, because there is a time delay also connected in the virtual platform, and I hear my voice twice with a time delay. So we can move to the next presentation by Dr. Vasilios Takavaklu, who is a senior researcher at the Hellenic Agriculture Organization, Demeter Soil and Water Resources Institute, who is also the innovation manager of AquaCycle project, as well as coordinator of Atlantis National Project in Greece, to which this presentation refers to. Vasilis, welcome, and please, the virtual floor is yours. You can share your screen and proceed with the presentation.
Thank you, Kostas. Thank you. I think that you can see my screen, yes? Kostas, can you hear me?
Yes, yes, we can see your presentation very well. Yes. So you can...
Great.
Yes.
I will start by saying that it's really nice to be in a panel among very good friends and colleagues, and actually it feels like home here. And since we are a family, let me take you to the sea, guys. These are numbers from the United Nations Environment Program, and these numbers speak from themselves. I mean, they speak and they say that Houston, we have a problem in Mediterranean. The densely populated areas around the coastal zone, the industry, the agriculture, as well as the human activities inside the sea, they all contribute to this problem. And this problem of pollution of Mediterranean Sea is getting more severe if we think that, if we consider that our sea is actually a closed basin. There are some openings, but the recirculation of water in our sea, it takes somewhere around 90 to 100 years. So what do we have to do? We have to do a lot. And to do it, we need solutions. As all the previous speakers before me mentioned, this kind of solutions can take several different forms, from political initiatives to public awareness, to prevention measures, as well as advanced technology for the treatment of pollutants. Well, actually, what you see here is a flooded wetland, and I consider them part of the solution, and actually a very interesting part of the solution. Flooded wetlands are actually a type of constructed wetlands. I will give a few information about constructed wetlands for our participants that don't know about these systems. Actually, constructed wetlands are engineered ecosystems, are engineered wetlands that they provide the services, the multiple services that natural wetlands provide. These may include from water quality improvement to flood water attenuation, or from habitat creation and enhancement of biodiversity to groundwater recharge. The trick in these systems is that they are engineered. I mean, we can play with the structural components of the system. We can select the plants that we are going to use, the substrate that we are going to use, or play with the hydrology of the system. In this way, we can maximize the service, the function that the system we want to perform, and that's how we target specific problems. Now, constructed wetlands are a very well-known system and commercially available with several applications. Floating wetlands are starting to get very popular in the last decades. They are used mainly in inland water, so they can be established in the middle of the lagoon or in a lake or in several other areas. And of course, there are also some examples of establishing floating wetlands in the sea. In most of the cases, we put them there to control, to filter sediments or to provide some habitats. These systems are, in principle, simple to create because we use a matrix that it floats on the water, and on that matrix we put the substrate for the plants, and on top of that we put our plants. After a while, we have a root zone floating freely underwater, and in this way we create an ecosystem that provides multiple functions. I mean, we can have water quality improvement through physical, chemical, and biological processes, as in case of the other constructed wetlands. And also we create habitats not only for the birds, but also for the fish fauna established in the sea. And now that you know almost everything about floating wetlands, let me introduce you to the Atlantis project. The Atlantis project is funded by the Hellenic Foundation for Research and Innovation. It's still underway, and the idea was to see if we can have constructed wetlands working for us and contribute to the pollution control in marine areas, and especially in the control of pollution that comes from petroleum hydrocarbons. In this picture, you can see a floating wetland that was established in the port of Ierissos as part of our project. These are small bioreactors in which several experiments are still taking place. These are actually, in these bioreactors, we use 300 liters of seawater in each one, and they are planted, the floating wetlands there, with 3 different kinds of plant species. I mean, we have Phragmites, we have Suaeda, and also we had Scirpus, because unfortunately Scirpus wasn't able to survive the conditions of experimentation. Although we don't have final results, but I would like to show you just a graph from our raw data, in which actually it's very promising, since the concentration of petroleum hydrocarbons was reduced in these systems in a percentage of more than 50% in just 3 to 4 weeks there. Now, what are the advantages of such systems in marine environment? First of all, as in all the constructed wetlands, they have a low cost for their establishment and considerable low cost of operation and maintenance. The truth is that comparing to other solutions, they can operate all year round, providing the service that we want in the system. And also, they can withstand the water level fluctuation and the sea waves. I'm not talking about mega waves there, but we are talking about waves that we can find in port areas or near fish farms or in any protected area where we can find a boat. Okay. Of course, they provide multiple other benefits, And this gives them the advantage to be very popular with the local population because they provide habitats for the biodiversity. They are greening the port area and also give an opportunity for educational activities. I mean, in our case, in Ierissos, there were several students visiting the site and learning about the ecology, the environmental engineering, and the environmental management. The opportunities are there. I mean, today there is a growing environmental awareness around society. And of course, there is a need, as you saw earlier, for reliable and cost-effective solutions. That's why we have this advancement of nature-based solutions. And this is a genuine nature-based solution, since constructed wetlands are able to provide not only environmental benefits, but also they have economic and social implications to the local communities. In this way, we have a favorable environment of opportunities for flooded wetlands, and these systems appear to be an attractive eco-technology able to address marine environmental problems in a sustainable way. At the end, I would like to look at the future a little bit. I would like to discuss about the challenges and the research opportunities. These technologies are live technologies. So first of all, what we need about flooding wetlands is the introduction of standards in terms of engineering and performance. Although constructed wetlands that are used in inland areas, they are well studied and they can be designed with precision. In case of flooded wetlands, the situation is a little bit different, especially when we are talking about a marine environment, and is based actually until today on the expertise of the designer. So we need to go a little bit deeper in this. Of course, there is a lot of opportunities to study and model the treatment mechanisms in the marine environment, the selection of vegetation, which is a very important issue, and actually it's a real challenge because it's a totally different thing to have a flooding wetland in the Mediterranean and a totally different thing to have a floating wetland in somewhere in the Baltic Sea. I mean, the amount of rain that receives the system is very critical in terms of the survival of the plants. And the Mediterranean environment, you all know that it's very harsh, especially during the summer months when the rains are very rare. Of course, we have the opportunity to check for the system effectiveness on emerging persistent pollutants, and also what's going on with the microplastics. Can we use it to filter some waters, or what kind of design we can use in these cases? Plus, the combination of the system with other systems and the introduction of innovative designs, as for example in aquacycling presented by Kostas, or the use of alternative biotechnologies, as for example the bioelectrogenetic wetland that I saw recently. These are all welcome solutions, and they can play in the game. As a last point, I would like to underline that we are in the COVID area, and the post-COVID area is in front of us. So we have to take advantage of the ICT technologies and use them in combination with our systems. They can provide solutions, they can help us in monitoring the effectiveness of our system and the location and the status of the system, especially in flooded wetlands where we usually don't have them outside of our door. It's usually out in the sea. So that was all for me. I would be very happy to answer your questions. Thank you, Kostas.
We thank you, Vasilis, for your presentation and for the new alternative approach that the opportunities provided by this floating wetlands. I'm sure Fabio will be intrigued as an expert in natural-based solutions by your presentation. He already posed a question in the chat, and I don't know if you have seen it. How do you deal with the plants?
Not yet, but I'm going to.
Okay. So, of course, this is a technical question. Although you mentioned this issue in the slide.
I would like to share my experience about this because it was a very interesting experience.
Okay, please.
When we started the design of the systems and reading all the references and the papers, we saw many interesting things. We saw that there are out there several species that they can survive these conditions, but actually in practice, it was something different. It was something different. I mean, from the 3 species that we had there, the Scirpus maritimus failed, couldn't withstand the conditions. Phragmites australis was somewhere around there. I mean, the height of the plants, Fabio, it's almost 50 centimeters, and that's it. And that's it. It doesn't go more. But Suaida maritima was the surprise for us. And if you would like to try this kind of things, Suaida, I suggest it. I mean, it survived, it thrived as a plant with a tremendous root zone system. I mean, it was great. And we had the best results so far.
So I see here a place for collaboration. So let's keep this idea for the future. I like also to stress that indeed also the European Union, as we have seen in the new calls, Horizon Europe calls, they stress the importance of coupling ICT tools towards water smart technologies. So there is a need for a disciplinary projects that brings also the engineers, chemical engineers, mechanical engineers, agronomists, and so on, with the ICT experts. Since the placement of sensors in monitoring the pollution and the current status of the water bodies is something that needs to be taken care of in the next projects. So, is there any other question for Vasilis? I propose to move on since we are already late with the schedule of our session. So I would like to invite Dr. Fabio Massi. He's a technical director in Iritra company engineering firm in Italy, which is nowadays recognized as a leading firm in the field of natural-based solutions and green infrastructure, of course, and a member of an Argument project also. And Fabio
Thanks, Kostas. And with a few words on, so we are also involving the use of conventional training, which will go further after some round tables and so on.
Recording in progress.
For the gray water is that the retention time in the system is a bit longer in comparison to other technologies like the vertical filtration in sand beds, which are the vertical filters. And therefore, we can expect for some elements which are quite slow in the biodegradation to have a better absorption and better removal in the time. And so we are talking about the compounds of concern, the sex. And the other technology that we are instead experimenting are these translation of the treatment wetlands on walls. Where we started working on this in 2015 was Indian project where there was no way to find horizontal spaces in the cities. And therefore, the only empty volumes that we were having for recycling the gray water was just on roofs or walls. And then we started developing this technology at that time in 2015. In general, in Europe, the technology readiness level of this technology, so recycling gray water by green walls, was about 1 to 2, so just pilot installations in the academy. Nowadays, we have gained some experience, and I would say that with a lot of research work, mainly done with the Polytechnic University of Turin, together with us and some other partners from Portugal, we have brought the TRL up to 5 to 7. So still the product is not at a commercial level or there are no, companies which are selling this kind of systems throughout Europe. Why to treat grey water is quite an easy task because grey water is the 75% of the domestic water used in an apartment. So if we are able to segregate it and reuse it closed loop, this is meaning that we will discharge much less water in the sewer system, and therefore we will have a big saving at the end of the pipes, at the end of the sewers, where we have the centralized unit treating the total water going there. The grey water is much faster in biodegrading in comparison to the mixed black and grey, what normally goes to the wastewater treatment plants, and therefore the kinetics for the degradation are much faster. You need lower retention time, and so segregating these 2/3 of the domestic water is really a convenient thing to do, and that should be considered everywhere in the world, because it's really a pity to lose this resource just mixing it with the other black water. So in the 8 pilot sites, we will have 5 of them, the ones which are bold here, which are exactly bringing to the realization of green walls with these amounts of surface each one. And so we will have installations in Italy, Tunisia, Jordan, and Lebanon. Obviously, we want to create some nice side effects, like, for instance, greening the urban setup, and even to create some microclimate good effects, as I've shown here. So the proven good effects, side effects provided by the green walls are even related to some purification of the water, of the air pollution, because you can have some interaction with the pollutants, the airborne pollutants, which are getting in touch with the green wall. And obviously the more easy in energy for cooling and heating the apartment, creating just a new insulation layer outside. Obviously, this will increase the biodiversity, which is the most impacted planetary boundary. So playing towards the increase of the biodiversity must be all times one of the first targets to get in mind. We can have even some reduction in noise, because you have some layers at different density to cross. And then obviously you increase the longevity of the building, because you are creating an external wall out of the wall itself. The aesthetics are not to be just forgot at the end. And so the role of the architects working together, the experts, the engineers, the chemists, the biologists, is all time very welcome. So we asked ourselves, why don't we even find a way to treat some water inside there? Because these green walls will have to be irrigated, so they can have a lot of nice functions in the cities, but they will need water. So treating grey water can be one way to irrigate them and keep the green alive, but then we get a very nice side effect, which is a new resource, the water produced in big amount. So we have published several papers with this super green project together with the University of Turin, and this is the first one. It's a sort of review, of general review, where basing on this one, we made the setup of the experiments, and then we defined the design parameters that we have used in the Now Ahmed project for designing the 5 installations in the 4 countries. So those are the more details. So in Italy, we will treat just a few water, 1.3 cubic meter per day by living wall. So the difference is in the 2 different ways to put the treatment on the walls are making use of these green facades, where you have the water flowing at the ground level and the plants which are just climbing over the wall, or otherwise you have to hang a lot of pots on the wall and keep the plants inside, and those are called living walls. So we will install a living wall in Italy, a green facade and a living wall in Jordan. In Lebanon, also both the units, and in Tunisia, just living walls, but a lot of them. So that's the highest, one of the highest flow treated by these pot-based units. So some design, particular of the design, which is not so easy to be translated in practice. Let's say from shifting from a 2 square meters pilot panel that we were having before to a full-scale installation is a bit of an engineering challenge. So you have to choose the right location for get a nice insulation. You have to give access for the maintenance. So it's not advised to go to hike on the front wall, let's say, because otherwise you will need, as we will say later, some more skilled personnel for making the maintenance. So here we have the details of the installations. So it was in Italy about 1,000 cubic meter per year recovered, the same in Jordan, the same in Lebanon, about 700 cubic meter per year in Tunisia. And what we have done is to try to translate the theory, let's say the background theory of what is applied to the vertical flow systems in terms of equations of prediction of performances, just thinking that every small pot, for instance, is exactly like a vertical flow wetland. And the same is reproduced here in the green facade design. The vertical wetland is this one. You just have it very narrow and linear, as we will see later. Obviously, grey water has some challenges, especially for feeding the pots or the ground channel. So as usual, for the treatment wetlands, you have to get rid of these kind of pollutants that can create issues in the feeding. So we are normally dealing with the suspended solids and with the grease and oils. And in this case, for the grey water, we need a pre-treatment, which is a simple degreaser. And then we need pumping stations to go up to the walls. And that's exactly what we have designed as primary treatments here. Then you have to decide the hydraulic profile, considering that at the end, you will have to store the water somewhere in order to reuse it into the houses for the closed loop recycling. And for reusing it in the houses in a safe way, we assessed already 15 years ago that the fecal contamination in the grey water is still very relevant. And so by these nature-based solutions, you can have some elimination of the microbial pollution, but you don't reach for sure the very safe levels that are required for reuse. And therefore, we had to add here a UV lamp for a final disinfection before the reuse. This is also already advised in some national guidelines where grey water is recycled, like the German one, for instance. What we try to figure out, trying to engineer, to shift to the real scale from the pilot plants, is just what we need then for making the operation and maintenance of the system. And so for sure, we have to deal with the degreaser and we have to remove greases from there. Then there are the regular checks on the electromechanical equipment to assess regularly if the plants are in good status or not, to remove the weeds in case they try to invade your green wall. And then one of the most delicate and appropriate things to do is to make a very regular checking, like twice per week, of the feeding and drainage systems. So the take-home message that we want to give here is to stay low. I mean, if you have to go up, then we will have to follow in a very active way what some researchers in Japan are doing. For instance, at the moment, they are developing some drones which are going up and down by remote command in order to cut the vegetation. But until we will have these drones for a very cheap price, then to work with this kind of skilled workers for the maintenance is still bringing at the end to a very high cost of the effluent to be reused, if compared to the normal cost of the water by the aqueduct. And therefore, you will not have a payback time short enough to convince private investors, families, and so on to install the systems. So as I was saying before, the grey water, the green facade are the simplest representation of the vertical flow wetlands. They are just very narrow ones. We have decided to keep a minimal width of 60 centimeters, and we are going to test if that's large enough for not having some effects, like, for instance, minor friction along the membranes which are waterproofing the trench. And so if you make a too narrow channel, then you will have this kind of sponge effect, which can shorten the retention time inside the system. And that's what we are going to check in these pilot realizations. For the pots, we needed to do a very long market research in order to identify modules which are good for us and still good for the plants. I mean, we want to have some hydraulic functions of each pot, which has to drip in the pot below in a very efficient way without discharging any drop of water outside, because this could create a lot of problems in terms of permits, let's say, and even safety of the people living nearby. And therefore, the selection of the modules is a very relevant step. And at the moment is also the one which is making the price increasing a lot in comparison to ground installations, like, for instance, the horizontal or the vertical wetlands. The selection of the media is another very delicate step. Lot of people, I mean, not so many, we are part of a green wall cluster in Europe, and there are at least, I don't know, 10 research groups which are working on the topic at the moment. At worldwide level, there are like 3-4 main poles where the research on green walls is performed at now. And most of the research done in the academy has been focused on the selection of the mix of media for having a slow permeation of the water in the pots, slow enough to have enough contact time, but even a very good absorption and a good surface for having a good growth of the biofilm, which is the main actor treating the water. So the mix of coconut, perlite, or leca, which is the lightweight clay aggregate, it has brought to very good results in almost all the different experiences worldwide. And what we have identified is that most of the existing systems are working with this range of oxygen transfer rate, where we have a mean value of 17 grams of oxygen per square meter per day, where in the literature, which is now about 25 years old, of the vertical flow beds, so thousands of papers published on those, we have a bit higher efficiency in the oxygen transfer, which is going from 32 to 64 grams of oxygen per square meter per day. So at the end, we have chosen staying in the middle of this range identified in the reported experiences, 32 grams per square meter per day as a design parameter for the NAWAMED installations. Same for the hydraulic loading rate, where the literature range goes from 100 to 750 liters per square meter per day, where normally the vertical flow beds are going from 80 to 120 for the black water, and up to 200 liters per square meter per day for the gray water. So we are bit higher in this case. We can give more water even if we have less efficiency in the aeration. We don't need to have so much oxygen at the end, because the ammonia level in the gray water is almost all time below 5 milligram per liter. So almost already with the right concentration for discharge. So there is no treat to make nitrification, let's say, and so the OTR is not the mandatory parameter to be considered in the design if you want to remove solids and organic matter only. The plant choice is instead quite important. Obviously, that has to be adapted to the local autochthonous plants, and we have to check several aspects, like how the plants are rooting, what are the aesthetics, what are the resistance to the climatic conditions, and not the least thing to check is if there is someone selling the plants in the sufficient number nearby without having to transport them from thousands of kilometers of distance, and what are the costs for the planting. So the best ones which have been selected in the project from us are these three: Lonicera, Carex, and Hydra. A very delicate issue is the feeding system, and with this slide we'll end up. For having a very good distribution, even distribution, all these parallel columns of pots is not a simple issue if you don't use the self-regulated drip lines, because they get to go to clogging in a very fast way especially in the first line, let's say, where the grey water is still very rich of nutrients inside. And so you can get some microbial growth directly in the feeding system and clogging of these valves, which is meaning that you have to use open holes. And for the open holes, then we need to regulate different branches with electromechanical valves. And for doing all this, we needed to make the use of the EPANET model in order to understand how to regulate the valves in all the time and in all the operational conditions. This will be presented in detail in the WETPOL conference next week. With this, I just want to show this nice infographic that we published like 5 years ago, where a lot of the elements that we are testing now in the NOVAMED project are just represented, and that's exactly the vision of the next smart cities and urban settlements that we have in mind, trying to develop and to innovate nature-based solutions for providing ecosystem services in the cities, like, for instance, the grey water recycling. And with this, that's all. I will appreciate a lot to have interchanges with the other water projects in the NECBC Med, because I see a lot of possible interactions. For instance, for the final disinfection, I've appreciated a lot what I've seen this morning. As also, I would like to work for the marine environment with the floating wetlands if really the plants are surviving to the 35 grams per liter of salinity. Thank you all.
Thank you. Thank you very much, Fabio. Really pressing your presentation with many technical details on the natural-based solutions. I really much appreciate these green walls and the vision of greening our cities that are full of cement is really something to, let's say, to hide and to seek. And we count on experts like you to push things, and especially stakeholders and those who take the decisions on what processes to implement at the local level. So any questions for Fabio, although we are a bit late and out of our time schedule?
A lot. Yeah.
Costas, one question.
Yes, please, Monfer.
Yes. What about the black wastewater? Fabio, you are doing a great job, actually. Congratulations. But what about the black wastewater? And have you noticed difference in odors between the horizontal approach and the vertical approach? Thank you.
For the black water, you mean?
Yeah, the black water. What about the black water?
Yeah, for the black water, where especially if you segregate it from the gray, and so you don't have these 2/3 of dilution created by the gray water, which has a very low concentration of ammonia, then all the ammonia will be in the black water, and so you go up to more than 100 to 120 milligram per liter, sometimes even 150 milligram per liter of ammonia. So for sure, you will need a vertical flow system, because otherwise, with the horizontal flow system, for getting sufficient removal, like 99% of removal of the ammonia for reaching very low numbers in the concentration, you will need really very, very long retention time, more than 12 days, most probably. I don't know now the exact calculation, but for sure, higher to 10 days of retention time, which is meaning lot of square meter per person equivalent. And that's not convenient anymore. You can do exactly the same treatment with 2 square meters of vertical flow constructed wetlands, reaching very good results. So the technology is nowadays very, very well known, and we are able to use models for predicting how the vertical flow will work in 20 years, let's say. So the engineering is very well developed about the technology nowadays.
Okay. Thank you, Fabio. I propose to move on since we have also with us Mrs. Irene Morel Rodriguez, Senior Expert on Environment and Climate Change of the Joint Technical Secretariat of the NCPC Program. And Irene, because we are out of the time schedule, if you would like to make a welcome speech and a short presentation of E&I, will be much appreciated. We can do it now. I prefer to do it now, not to keep you so long in our session. Hello, Irene.
Hello, Kostas, and hello, everybody. It's a pleasure to meet you also in this context, and I'm very pleased to hear also the presentations because they have a good quality, and it's very interesting to hear also about all these interesting initiatives. I have made a small presentation, just 5 slides.
You can share your screen.
I will try to share. Can you see it?
Yes.
Good. I will try to put it in
full presentation.
Presentation. Okay. Can you see it well without cuts? Yes?
Yes.
Okay,
excellent.
Good. Well, it's a small presentation also for those who are not aware about the program NECPC Med. It's the European Neighborhood Instrument, and it's one of within this instrument is the only program that made cooperation within the Mediterranean basin. The program has 4 thematic objectives and priorities, and those are developed in two priorities that are the promoting of economic and social development and address the common environmental challenges. 3 of them are on this first, and then in the environment, we have 4 categories that are water efficiency, waste management, energy efficiency, and integrated coastal zone management. Here you can see a picture about what is the area of cooperation, because the Mediterranean, as you know, it's a very complex place, but also shared by everyone. So it has the north, the south, the east, and the west with different challenges, but also the solutions are always in getting them together. So here you can see also the places where and the regions marked in blue where the program is developing, and also the eligible and the adjoining territories, but also the places where the project would also try to do also more like Algeria, which has signed the program, but is still like in the financial agreement. And there would be also other places where the program would be held. To make a small wrap up, I wanted also to tell you that the joint operational program was adopted in 2015, and this is the program from 2015 to 2020. There will be a next program that will be, it's also under development and will be implemented in the future. Now there are 14 countries involved, 209 millions, and it's managed by the autonomous region of Sardinia, which is in the middle of the Mediterranean, and also providing support to all the partners. Here can you see also again the 3 for the first on promotion of social economic development and the common challenges in environment. To give you and provide you a little vision on the calls that have been done, there is one call that was the standard call with 41 projects, and this is in those that the projects that were presented today are included. that are at least of mostly 36 of 3 years of implementation, and 41 projects were selected. Their budget depends, and it's from 2 to 3, that depends, millions. So they are big cooperation efforts. On the strategic side, they are other kind of projects that were selected later and that have more a vision on strategic issues. 23 projects were selected, and they are already putting them into force. They started last year, and they are only for 2 years. Lastly, there was a capitalization call that was done recently, where 11 projects were selected regarding the budget that was available, 11 million also, because each of the projects has mostly 24 months and has 1 million, around 1 million of budget. But there was also an extra apport that made the possibility to have 5 new projects that were also on the list and could also be selected and will be also now being implemented. Oh, I have this in Italian. Yes, we have 80 projects now in force. This is the amount of all the projects, and here you can see by each priority how many projects do we have for each one of the calls. For the environment and climate change, we have 17 standard projects, 2 strategics, and we are very glad that now 6 capitalization projects have been accepted. Okay, just to mention about the synergies, the exchange of knowledge, and the good practices in the program. We welcome these initiatives. I'm very glad also to see that the presentation on the living labs, which is one of the tools that the program is using, and it's very interesting to foster science and community, because it's one of the issues that is always very difficult to engage society, science, results. We always talk about this, but it's always difficult to find real good examples of these. And here also the session of today was very interesting, because all the projects for the priority 4.1, innovative solutions for water management, could present the results. That is also very interesting, because we are talking about science, putting them into the field with initiatives that are on the ground, that give to the communities a real vision of other solutions, and that, as you see, they are already, even if the projects are not finished, they are in an advanced phase of implementation, where the results are already there. The 5 projects that are in this initiative, Aquacycle, Metis, Menawara, Nawamet, and Prosim, they are exchanging and having a good exchange between them. So this is also a possibility to have a better result, not only the one of one of the project, always also to exchange, to know what other colleagues are doing, and also this exchange from north, south, east, and west is ensured, because also we see that the partners, they have their own dialogue, and this is how we can also foster new projects, new future, and that initiatives could be also achieve not sustainability, but at least a long period, not only for the project life. I let you hear also, for people who didn't know a lot about the project, some of the interesting links for the platform, where you can see the projects, where you can know them. And you can find also in the info center some news about calls, some news about opportunities, also about jobs, because many of the projects offer specific calls for tenders and for specialists. This is also very interesting to know about it. And it's interesting also to see that on the practical side of the projects they are alive, but also you can contact them. Also today you saw most of the project on the scientific side, on the technical side, to contact the persons also in the cluster to exchange, because all the projects are open to this. And also it's very interesting. I saw also that the presentation about the floating
wetlands from
wetlands,
project,
yes, very interesting. I know that also in Tunisia they are also developing a lot of plants, and they are adapting plants to salination. So there is in Borj Cedria an institute. I think there you could also find interesting places there to find more species for these islands and make them more biodiversity. Yes, yes, very interesting also to see this news and that the science is also turning to the ground. Well, that's all from my side. Thank you, everybody, and also to be here.
Thank you very much, Irene, for all your support all these 2 years. We really must appreciate as a cluster the support and coordination provided by the Joint Technical Secretariat. As you already said, this cluster was meant to exchange ideas on how to share and enhance the impact of our project activities and results. And our common inspiration is to find innovative and technological solutions to increase the water efficiency in the Mediterranean region. Thank you once again for your presence to our special session. And I would like to invite Mr. Hamadi Kalali, senior researcher in CERTE, which is the Research Center of Water Technologies in Tunisia, and member of Aquacycle project, to present his work on the SWOT analysis of the institutional policy and regulatory framework governing wastewater treatment and reuse in Tunisia, Lebanon, and Spain. Please, Hamadi, the floor is yours.
Thank you.
You can share your screen.
It's okay.
Yes, you can put in full presentation mode and we can start.
It's okay.
Perfect.
Okay, thank you all for CEST and Placas for to give me this opportunity to give a presentation on what we have done till now on the work package 3 of Aquacycle. What it is a SWOT analysis of the institutional policy and regulatory framework governing wastewater treatment and reuse in Tunisia, Lebanon, and Spain. I will give this because I don't know when I am scheduled, but this is in the framework of Aquacycle, which you have presented earlier. So the approach to the SWOT analysis is collecting both positive and negative aspects concerning wastewater management by using a systematic approach in the 3 countries where the pilot APOC pilots will be constructed. The approach is one, it's documenting the context based on a desk review, and 2, collecting viewpoints through questionnaire and interviews with stakeholders. The documenting context, it's to answer the questions: what are the national policies? Who are the actors involved? What are their roles and responsibilities? And which projects or plans have been initiated? The answers to these preliminary questions were obtained through a desk review in the 3 countries. In the national policies for the outcomes of this desk review, in the national policies with respect to wastewater use, first in Lebanon, we saw that there is no legal framework in place, and the use of untreated wastewater is prohibited by only a decree issued in 1911. In Spain, there is since 2007 a royal decree establishes the regime of reuse and allowed uses, and all the monitoring and the procedure for obtaining the grant of reuse. And now in Spain, mainly in Murcia region, they are full in process to comply with the EU directives. In Tunisia, since 1989, there is establishment of national standards which regulate on releases of treated wastewater and the environment and on the use of treated wastewater in agriculture. And there is a list of crops that can be irrigated. This list was established by the Ministry of Agriculture in 1994. For the projects of treatment plants in Lebanon, we have many wastewater treatment plants in projects, but few are implemented. In Tunisia, we have 122 wastewater treatment plants according to ONAS report in 19. So we have urban wastewater treatment plants and rural wastewater treatment plants, and some industrial wastewater treatment plants, which collective industrial wastewater treatment. In Murcia, we have many also wastewater treatment plants in Murcia region. We have here the spread of this wastewater. We know that almost all cities and all regions, localities are fed with wastewater treatment plants. So the next step where it's to collect additional inputs through stakeholders questionnaire. The script for the questionnaire and interviews is an insight about wastewater management stakeholders' perceptions, views, plans, activities, suggestions, and the willingness and readiness to adopt the ABOX system. A series of in-depth structures interviews were used based on beforehand dispatch of questionnaire to the interviewers. And they are after discussed face-to-face and finally transcribed. 4 themes are discussed. What is the current status of domestic wastewater treatment, which included states of sanitation, main barriers that hinder the sanitation needs, availability and performance of operating wastewater treatment plants, sanitation changes challenges in small communities. The second theme is what is the current states of treated wastewater reuse, the strategic regulations and norms in place, reuse management, wastewater reuse plans at regional, national level, perception of wastewater reuse, criteria, drivers that support or hinder decision for wastewater reuse project. And the third, how effective are initiatives with regard to those aspects and advocated steps. The fourth, it's willingness and readiness to adopt the APOC system, mainly for knowledge about the system and the training needs. We implement 15 interviews in the 5 by country. And the results of the SWOT analysis is for Lebanon, for the strengths that in the Tripoli region where you will implement the APOC system, there is one treatment plant which is considered as one of the largest plants in Lebanon. But for the weakness, there is an obsolescence and overload of treatment plants result in water quality outside of the norms for safe reuse. There is a lack of financial resources for OPEX and CAPEX. Institutional gaps behind the poor sanitation in rural areas also. For the opportunities, as the field is open, so we are there is opportunity to adopt a new paradigm for sanitation in mid-sized cities and rural areas, and to adopt new technologies also to sustainable development, such as the APOC system promoted by Aquacycle. For the threats, there is many loss in financial institution trust in debt repayment, and corruption in allocation of public contracts, and delays in projects implementation. With the financial now crisis, and it will be some threats. The second, for Spain, there is high level of experience in current wastewater treatment systems, a lot of studies and research to improve the viability of the treatment, to explore novel systems and advances in food safety. From the weaknesses, we notice that some funding depends on political decisions, not technical, and the consumers are not always confident to buy agricultural produce irrigated with reclaimed water. As opportunities is also to develop a new system which is more environmentally friendly and with lower operational costs, to go further in the study of emerging compounds and their impacts, to sensitize politicians on the need for further research. The threats, it's always the lack of future funding for research. For Tunisia, the strengths, there is availability of treated wastewater for use. So we have 200, now it is nearly 300 million cubic meter per year is treated to level 2. And there is a program for enlargement of wastewater treatment to encompass tertiary treatment, so level 3. From the weaknesses, there is a mismatch. We note there is a mismatch between treatment plant locations and potential reuse areas. And there is also, as in Lebanon, obsolescence and overload of treatment plants, which results in water quality outside of the norms for safe reuse. And there is a lack of financial resources for APEX and CAPEX. For the opportunities, we can also now, for the spreading of the wastewater treatment plants in rural areas, we can adopt also a new paradigm for wastewater reuse and adopt also the new technologies to make the quality of water adapted to the reuse. For the threats, there is also, with the political crisis in Tunisia, loss of financial institution trust in debt repayment, corruption also in allocation of public contracts, and many delays in projects implementation. So the strategies what we proposed to policymakers, which made by the Aquasite partners, is that as the sector of sanitation and water treatment plant is not fully developed in Lebanon, it is time to embrace a new approach and a new paradigm to tackle the problem with a vision based on closed loop approach. A diagnosis of the actual situation of the concerned sectors is necessary, as well as the identification of coherent technical, economical, and institutional options to rationalize investments in the field. For this purpose, it is imperative to involve and to engage stakeholders in the establishment of strategies for a sustainable sanitation in the country. So if we want to wrap up, it's to achieve a deep diagnosis of the causes of the poor sanitation situation, to identify a coherent, also technical, and for the future investment, to organize awareness-raising campaigns aimed at changing citizens and especially farmers' perception of wastewater reuse. For Spain, the region of Murcia is on track to achieving full compliance with the EU Urban Wastewater and Water Framework directives. So now is the appropriate time to share the expertise with other autonomous regions, as well as beyond the borders of Spain, like Tunisia. Continue investing in research and novel treatment technologies, not least to deal with emerging compounds, of which the impacts on society and the environment are not as yet fully understood. We saw with Imane that this is ongoing research. In Tunisia, we saw that Tunisia has extensive experience and a long time tradition on wastewater treatment and reuse. But the SWOT analysis showed that today is the appropriate time for renovation and upgrading of wastewater treatment plants existing, and revision of water reuse standards, more coordination between public stakeholders, more information sharing, for example, accelerate the establishment of the National Information System for water, strengthening procedures related to the involvement of farmers, diversifying the monitoring indicators and actors, and an economic reflection to review the pricing structure of the use of treated wastewater, but also to identify promising markets, and a multilateral exchange between research institutions. So if we wrap up, it's stepping up efforts aimed at safe reuse of treated wastewater through improved coordination between public stakeholders, strengthening the involvement of farmers in the reuse action plans, and review of reuse standards and of pricing structure. So we saw that is the conclusion of this SWOT analysis, but we have also a common conclusion for this SWOT analysis for the three countries. The three countries, the governmental authorities are called to put the following required strategies: build partnerships with community, private sector, and support organizations, decentralize the sector, increase the democratic process in decision-making and formulation of strategies of the sanitation, wastewater treatment, reuse sector for cities, enhance bottom-up planning by generating community-based initiatives, increase the government responsiveness, enhance comprehensive, appropriate, and logical communication between government and community, encourage community-based organizations and micro enterprises for the sector, mainly for rural areas, and look for mutual consensus among the stakeholders for the best solution and appropriate strategy. So the next steps for this SWOT analysis, we performed the first series of stakeholders workshops in Lebanon, Spain, and Tunisia, with the theme changing the paradigm for wastewater treatment and reuse. We look forward to present the outcomes of these stakeholder workshops in another occasion. And now we are planning for a community approach to sanitation wastewater treatment and reuse for the second series of stakeholders workshops in Lebanon, Spain, and Tunisia. So and finally, I want to have the acknowledged. So for the Aquacycle project and the NECBC MED program, and as a disclaimer. Thank you for your attention.
Thank you, Hamadi, for your presentation and in summarizing the SWOT analysis carried out in the context of Aquacycle projects in regards to the institutional policy and regulatory framework in the target countries concerning the wastewater treatment and reuse. I'm sure that the coordinators and representatives of the other projects can acknowledge the results of this SWOT analysis, and we will be much appreciated if they have any comments or they would like also to put their knowledge on this SWOT analysis to our outcome. They are much welcome to do so. So I propose to proceed, if you agree, to the next presentation by Monther. Monther, last but not least.
Very much.
We are really much looking forward to listen to the news of MEDIS project. Dr. Monther, he is the General Director of the Palestinian Wastewater Engineers Group and coordinator of MEDIS project. So Monther, the floor is yours.
Well, thank you very much. Well, I will not be talking actually about MEDIS. I will be talking about another project.
Ah, okay.
Do you see our slides?
Yes.
Yes, good.
Yes, yes.
Let's say we'll be talking about another actually project which we have implemented, having to do always with non-conventional wastewater. This is just to bring a change to the program actually. And the Palestinian Wastewater Engineers Group has succeeded in implementing a comprehensive wastewater project covering completely entire village located in the Jordan Valley called Marj Al Ghazal. It is a decentralized wastewater management and reuse scheme in this arid area of the Jordan Valley. We contributed to the improvement of access to non-conventional water resources, improving health, economy. We supported food security, protected the shallow aquifer. Actually, to my knowledge and to my information, it is the first project of its kind where we succeeded to completely give full wastewater services in a decentralized way to an entire community. We have to achieve that, we have planned and implemented a lot of activities within which the construction of 11 collective household grey wastewater treatment plants and 11 modified septic tanks, giving wastewater services to 47 households in which we have 228 persons living in these households in the entire village, and giving services, also wastewater services, to the village school where 150 students from the village and nearby villages are studying. When I say collective household grey wastewater management, I mean that we have gathered few houses, up to 5 houses, in one wastewater treatment plant and in one modified septic tank. They are sharing the same treatment plant operation and maintenance, and they are sharing the reuse of the treated water, using it in irrigation of the nearby palm date orchard. We have succeeded in collecting 34,200 liters of grey wastewater, treated grey wastewater per day, using it completely in the irrigation of 13.5 dunum, 13,500 square meters, where 135 mature date, medium age mature date are irrigated, in addition to the irrigation of 2 greenhouses of 300 square meter each. Let's say when we say medium age tree, it is a palm date tree which is producing 80 kilogram of palm date per year. We have been doing the economical analysis, the benefit, the socio-economic analysis of this action, noticing that we have revenues and savings. Of course, we have also expenses, but in addition to the health improvement, to the let's say environmental protection, we also obtain via this project some economic benefits. So making the analysis, the financial analysis, we noticed that we have saved 3,212 euro per year as cost of irrigation water, because we have 12,483 cubic meter per year, and this, if the farmers would buy this quantity of water and use it for irrigation, they would spend 3,212 euro. This amount of water, when it is used for the irrigation of the palm date orchard, it is having a benefit of 27,324 euro when we sell the almost 11,000 kilogram of mature date yearly. As I said first, each tree produces around 80 kilogram of date, and we are selling each kilogram at the cost of around 2.53 euro per kilogram of mature date. Of course, the same tree is producing dates which are sold at 10 euro per kilogram, because there are different categories of the same date produced by the same tree, where there are what we call super jumbo date, with one fruit weighing 23 grams, and there are jumbo, there are large, medium, small, etc. In average, we have 2.53 euro per kilogram. We have also revenues from selling the vegetables cultivated in the 2 greenhouses. We have an estimation of 16,000 euro per year selling the fruits produced in the 2 greenhouses. To have these revenues and savings, we have to spend on the operation and maintenance of the 11 grey wastewater treatment plants. We have estimated €3,000 per year as cost for operation and maintenance of these plants. And we have also to maintain the orchards and the greenhouses. The operation and maintenance of these are estimated at €5,468 per year. Of course, these two expenses are made by the same beneficiaries of the greenhouses and the treatment plants. Nevertheless, we have opted to have an estimation as if these works are made by extra people, by skilled and non-skilled workers, as if they are outsourced. Looking to this table, we have more detailed information about the economic benefits of the project, where 47 households of the whole village, in which there are 228 people living, they produce 12,483 cubic meter per year of treated grey water. And this cost, if they would buy it as irrigation water, then they would spend €3,121, considering a cost of €0.25 for each cubic meter. These are used for the irrigation of an open area and a June palm date orchard of 13,500 meters, and in which there are 135 cultivated trees. Here we say that each tree gives 80 kilograms per year, with a cost of €2.53 per kilogram. So we have a total revenue from selling the dates, the fruits from these trees of €27,324. In addition, we are using this treated grey wastewater for the irrigation of 2 greenhouses of 300 square meters each. And these are producing an average of 60 kilograms of vegetables on daily basis, selling each kilogram of vegetables like tomato, eggplants, beans, and so on at €1 per kilogram. And usually I have to say that it is sold at a higher price because it's an early matured vegetables being cultivated in protected environment, having between brackets intensive agriculture, being cultivated under greenhouses. We have a revenue from these greenhouses of €16,000 per year. Part of these vegetables, in addition to what the farmers are selling, is consumed at the household level. There are the two costs we talked about, €3,000 for the operation and maintenance of the grey wastewater treatment plant, including a minor part used for the cost of electricity, because we have a small pump inside the low-cost and low-tech treatment plant itself. Operation and maintenance of the orchard and the greenhouses is €5,468. So we have a total net revenue of almost €38,000 per year. If we divide this revenue per the households in the village, then each household is having something like €808 per year. In addition to the saving which they have on emptying their septic tanks, we didn't estimate, we didn't take into consideration a cost amounting at about €16,000, which is yearly spent for emptying the septic tanks, which now they don't have to spend anymore. But these are some pictures which sheds the light about what is being done. This is the crew of the project. This is a photo of the treatment plant, one of the treatment plants we have constructed, and this is a photo from up, which you can see to the modified septic tank, where 20% of the wastewater produced at the household level, which is the black wastewater, is treated in. This is a photo inside the greenhouse, where we can see a lot of tomato is harvested. On the social level, let's say, The project contributed to the improvement of the social relationship between the village residents, actually. In addition to improve their health and well-being, we actually could not measure in precise figures the improvement in health conditions because unfortunately we didn't have initially a quite precise and enough information about the prior existing health conditions of the village, but all village residents, they stress the fact that they have less frequent visits to medical centers. Of course, the project all declared in the questionnaires we have distributed and in the given interventions and interviews we made that their social relationship with their neighbors has improved. They have less quarrel, they have less conflict, there is no more wastewater flowing from neighborhood to another neighborhood, there is no more problems of bats, insects, and so on. Well, this is all from my part. Thank you very much.
Thank you very much, Monfer. It's really very interesting, your presentation, and very detailed and concerned to the socioeconomic analysis you carry out with the proposed, let's say, solution for treating grey water at a local level. I'm very much impressed by the analysis you carry out. I'm sure the same is also for the rest of the participants. I have many questions, but I don't have now the time to do so, to tell you the truth. I understand that you count on anaerobic and aerobic treatment by taking advantage also natural trees, for example, to carry out this aerobic treatment after the anaerobic part. I don't know if there is any short question for Monfer or for any other of the participants. Sorry for keeping you 1 hour longer than expected, but I find that our session was very productive. This was actually the first event carried out by the cluster of the water cluster of ENI, funded CBSMED programs. And I'm really looking forward to a new event in the next months to come. I'm very glad for our collaboration. I'm also looking forward to our new Medway Cup project. I would like also to attend your attention that there is also now at 6 o'clock a round table organized by the conference organized by the Natural Environment and Climate Change Agency for those who would like to attend. So thank you very much once again. I wish you all the best. Please stay safe and negative from COVID, but positive, of course, as September. So have a nice afternoon, and I'm looking forward to our next joint event. So bye-bye to all.
Thank you, Kostas.
Bye, Kostas. Thank you.
Nice evening.
Bye.
Bye-bye.